My name is Michael Gottesman. I'm the Deputy Director for Intramural Research at the NIH. And it's my pleasure to introduce this session, which is the third workshop in a series at NIH focused on reproducibility of data collection and analysis. The first session was on cell biology, the second section on structural biology, and this one, of course, is on genomics. This is an area of enormous interest and importance in science. But sometimes the analysis of the data and the interpretation of that data are quite distinct phenomena. So without spending too much time by way of introduction, I'd like to introduce Eric Green, who's the director of the National Human Genome Research Institute. Eric was an early adapter for genomics. He was a genomicist before genomics was in style, before anyone could even pronounce genomics. I remember the days when we had all kinds of interesting pronunciations. And he brings his enormous energy and knowledge to this field, and is going to make some comments about the great strengths of genomics and also some of the problems that can befall us if we're not careful. So, Eric.
Thank you, Michael. Let me add my own welcome to all of you, some traveling from elsewhere to come in and join us today. It's greatly appreciated. Let me say, I think this is terrific that we're having this series. I applaud Michael and his team who have been organizing these gatherings in different areas, as you've heard. And it really does reflect the fact that NIH is taking very seriously some of these issues surrounding reproducibility in biomedical research. This does get discussed frequently, actually as recently as this morning at the Institute Directors meeting. This came up about how important this is and how this is clearly something that NIH is going to pay very careful attention to, as are other funding agencies in other countries who are equally now getting engaged in this and actually in dialogue with NIH in very productive ways. As Michael alluded to, this is the third of three major areas that are being looked at, although I would contend, and I think it's accurate to say, this is sort of a field of science that's relatively young. He was sort of making reference to genomics and my age, and I'd like to think of myself as young. I'm not as young as I used to be, but the truth of the matter is the field of genomics is really only about a quarter century old. Depends exactly where you put the starting line. The word was first coined and put into the scientific press in an inaugural issue of the journal Genomics that was published in 1987. Some people mark that as sort of the start of the field. Others put at the starting line as the start of the Human Genome Project, which was sort of the iconic beginning of the field of genomics. And we're coming up, by the way, on the 25th anniversary of that moment. October 1 of this year will, in fact, be the 25th anniversary of the launch of the Human Genome Project. In thinking about the field, I get asked to speak about genomics a lot and often point out sort of the raging successes of the field over the last 25 years, whether it's the significant, impressive reduction in the costs of sequencing DNA, nearly a million-fold now since the beginning of the Human Genome Project. Obviously, the Human Genome Project itself as a raging success. Seeing the new tools of DNA sequencing and the technologies applied in productive ways over the last 10 years in particular to really amass large amounts of information about human genomic variation. We now know nearly 100 million sites in the genome that are variable in the human population, and that number continues to grow. A number of efforts have been focused on that. And then increasingly, over the last 12 years since the end of the Genome Project, we have learned more and more about functional sequences in the human genome and the choreography involved in having those functional sequences orchestrate human biology. And then finally, of course, and I'm sure some of that will be touched on even here, we are seeing this now move from a very basic science endeavor increasingly into a translational research endeavor, and now very exciting opportunities to make this a clinical research and eventually a clinical care endeavor. And we are seeing vivid examples of genomic medicine emerge and be clearly important for clinical care. And that, of course, is just the tip of the iceberg, which is so much about what my institute, NHGRI, is focusing on. The other accolade I think genomics does deserve, although I know this is going to get unpacked more throughout this workshop, is I do think we deserve a considerable amount of credit for being leaders in thinking about issues around reproducibility, data quality, and so forth. And I was sort of thinking about, well, why is it that we sort of have done really quite well for a young field in this particular area. I think some of it's the nature of the data, the nature of it's not it's very, by its nature, a lot of it can be measured quantitatively, and it's very digital in nature, and I think it sort of lends itself to circumstances to really be able to scrutinize, develop approaches to make sure things are reproducible. I also think it's the nature of the projects that have sort of been emblematic of the field of genomics. You started the Human Genome Project. It was very consortium-based, and a lot of projects that have followed, whether it's 1,000 Genomes, the ENCODE project, HapMap project, and so on and so forth, and I could sort of list out many, many large consortium-based projects. I think because of the nature of that, there's a lot of interactions with a lot of investigators who are asking each other really hard questions along the way and are sort of looking over each other's shoulders, asking questions when things don't reproduce, even within a consortium. Builds in a lot of checks and balances sociologically, I think, that has contributed to it. I think the other aspect that's contributed is within the culture of genomics, of course, has been data sharing as one of the underpinnings of the field. It started with the Genome Project, but continues to the present time. And I think with the knowledge that that data you're generating is about to go out the door pretty quickly and be looked at and scrutinized by others, I think sort of brings an awareness that you want to make sure that data is good and that it can be reproduced and so on and so forth. So it really has created a focus on quality that I think sort of runs through the whole community. Maybe you don't realize this. I'll just give you one example. Some of you I do know realize because you participated in this. But during the Human Genome Project, there was a phase where they wanted to make sure that what they were stating to be the quality of the sequence being generated was indeed that quality. And so the consortium of investigators involved in the Genome Project started to basically do a round-robin comparison where they would send clones that had been sequenced at one site to another site, have it be independently sequenced, and then compare to make sure that the sequence that was generated at site A reproduced to be the same sequence at site B. And in doing so, actually, the good news was that they discovered that the quality of the sequence they were generating was tenfold better than what they had actually estimated or what they had claimed. And so I think, again, and then similarly in projects like the ENCODE project, Encyclopedia of DNA Elements, exquisite attention to data standards, establishing data standards, and thinking about making sure that what is being generated with some of these new fancy technologies would be the same from site to site. And then the last example I'd give, I think, in one of the most recent examples is the 1,000 Genomes Project, Human Genomic Variation Project. Really, in many ways, really contribute in significant ways to developing lots of analytical methods to make sure that sequence data generated with the next generation sequencing methods, all these new sequencing methods, indeed that it was reproducible from site to site, and that you could believe it when somebody generated sequence and said, this is a variant, it's not an error, it really is a genomic variant. And I think they really have elevated the sophistication level with respect to how those kinds of studies are being done. I give those as examples to tell you one more story before I wrap up my comments. I don't know how long ago, maybe about 10 months ago, maybe a year ago, again, with a lot of attention going to reproducibility, Francis Collins requested that every institute put forward a pilot project in the arena of reproducibility, that every institute had to come up with something to somehow sort of demonstrate or sort of actually have a real exemplar circumstance that could be pointed to where you are testing in your field of study reproducibility. So we could learn from that. And I went back to my staff and I said, well, what should we put forward? And mostly the reaction I got was, well, almost everything we're doing, and sort of listed off all these consortium and all these things we are doing, and we had been doing for a long time. And so, and there wasn't really much interest in doing something beyond that because we thought we were actually already overachieving. And so that was what we submitted. We said, you know what, we're sort of already doing this. And we would prefer not to do it because we would point to many of these examples in many of these consortium. And we got the thumbs up that we sort of got advanced placement credit, I guess as you'd say, where we got a pass. And so again, I give that as an example because I think it's a reflection of the field as a whole, that I really do think genomics has been a leader in this, and we even were able to get out of a homework assignment as a result of it. But going forward, I don't want to lead you with the impression that I think we're fine, we're done, we don't need to be thinking about this. It's absolutely just the opposite. I actually think that we should remain and should endeavor to remain leaders in this. And part of the reason I think that's particularly important relates to sort of the democratization process going on with some of the technologies inherent in genomics and the widespread use. You know, without question, these technologies that are being developed come with lots of analytical challenges associated with them. And increasingly, of course, what we're seeing is these technologies are just spreading across the biomedical research landscape. I see this at NIH all the time because there's genomics going on at every institute, certainly not just our institute. And those genomic approaches are being used by big labs and small labs and individual postdocs, individual graduate students, and so on and so forth. I think that, therefore, comes with a sort of an extra responsibility. It's no longer big science. It's no longer just consortium science with lots of inherent checks and balances. Rather, we need to make sure that as these technologies infuse out to smaller groups, individual investigators, that these same checks and balances are sort of built into the methods or built into the routines or the approaches. And so what I would sort of charge this group to do, and I think you're going to do as we think about it, is to sort of help NIH and help the genomics community think about how to continue the high standards we've established in the first quarter century of the field and make sure that those same high standards apply as genomics becomes a routine tool in many, many biomedical researchers' studies. And I think that's the kind of thing that can really be facilitated by the gathering today, the talks that we're going to have and some of the discussion. And I would really, once again, applaud this gathering and encourage you to think about what could NIH facilitate in this area to make sure that just as much as the genomic technologies are being spread across the community, that similarly these routines for high standards, high quality, and reproducible routines spread with them in parallel. So with that, I will stop, and I'm going to turn this back over to Michael. Thank you very much.
Thank you, Eric. I just wanted to say a couple of words about the format of the workshop. There are 4 sessions. Each session is moderated by a major editor of a major journal in the area of genomics or genetics. And we have a slight change in the schedule today in that the 3rd session on ChIP-seq and other epigenomic tools will be the last session. So that will be, instead of 3, will be at 4. And the 4th session on RNA-seq will be at 3. So other than that, I just want to remind you that this is being broadcast live. So if you're asking a question, make sure you go to the microphone and make sure that what you say should pass normal diplomatic muster. And it will also be archived, so you can go back and listen to yourself speaking if you'd like. And many other people will be listening in. There's a rapporteur who's making notes, and the speakers will have a chance to edit those before they go public. And this is intended as a resource for months, certainly years to come, for people who are interested in entering this field or who are in the field who want to know more about specific technologies. So with that, let me introduce David Nelson, who's the moderator of the first session.
All right, thank you, Michael, and thanks to Paul for the invitation. I'm really excited to be here this afternoon and happy to help with this session, which is I guess reproducibility in clinical genetics and genomics. And I've included the word genetics because I think that's really the foundation of what we're going to be talking about for the next hour. So how do I advance a slide here? Reproducibility is a topic that has been important to geneticists from the beginning. You know, we have a long history of accounting for reproducibility issues in genetics, and those really go back to Mendel, who did have some problems with reproducibility in his experiments with peas. And so we've developed terminology in genetics for the last more than a century to address this variation in reproducibility, things like incomplete penetrance, where the same allele doesn't cause the same phenotype in 2 different individuals, variable expressivity, where you have different levels of effect of that allele, allelic heterogeneity, where different alleles in the same gene give vastly different disorders, locus heterogeneity, and genetic modifiers, obviously. People are a little more difficult than your usual biological system to study. Biological systems are inherently messy. There's there's a lot going on in terms of chaotic events, and in human genetics, it's particularly difficult. Despite our spectacular phenotyping, our ability to really burrow in and understand a person's phenotype in great detail, we're limited in the genetics of studying individuals by existing variation, whether that's common variation or rare variants, as we'll hear about from our next two speakers. The influence of genetic background, obviously, is something we have a difficult time controlling. We can't control environmental influences very well. And naturally, we can't set up the breedings the way we'd like to get the results we want. We can't create alleles in humans. We can only study what's pre-existing in the population. In other species, of course, we can control these aspects much better, but then the phenotyping lags. And as someone who works with mouse behavior, I'm well aware of the difficulties there. So we're going to hear first from Steve Chanik, who's director of the Division of Cancer Epidemiology and Genetics, about common variation and demonstration of association. And then next from Les Biesecker, who's chief and senior investigator of medical genomics and metabolic genetics branch and head of clinical genomics in NHGRI. And I think the theme here is causality. We're really interested in understanding, does that variant really cause that phenotype in that individual? What evidence is sufficient to support that association? How do we use those findings ultimately to understand disorders and treat disease? So I'll turn it over to Steve at this point.
Well, thank you. It's certainly a pleasure to be here, and thank Michael and Eric for their I think very important and very prescient remarks about the context of today's discussion. I think that it's very timely and one that we probably should put on the calendar to regularly review over time as we see the extraordinary changes taking place with technology, and particularly, as Eric referred to, the democratization, which I think is really the key issue. As these things move from a few central places to many, many people doing it, it really changes the way in which the science is conducted. And I also really appreciate Dave's remarks right there at the start of how messy it is in the human system. So my comments are going to be primarily directed towards the challenge of humans. Now, so I'd like to start by really saying I think there's several things. There's an arc of genetics that we really have to think about. We go through stages. Very, very rarely are we in a position to say that the first observation is truth and that we're absolutely certain. We live by this idea of reproducibility in terms of the integrity of the data, but also in terms of being able to see data in multiple iterations repeat and replicate something. And so I think we have to be really careful, particularly when we think about the clinical and the translational aspects of this, that we can't get ahead of ourselves too quickly. And I think there are a number of technical and practical issues that really are important in thinking about this. So my talk today is going to be primarily focused on the discovery element, because it's the discovery that's the etiologic foundation that then we can take to the clinical setting and ask the important questions in the clinical setting, where, again, the messiness of human biology is such that you can't necessarily say this variant is going to immediately translate into an actionable circumstance or what those recommendations are without studying. I also think we have to think about some of the myths that are out there, and particularly for those who are thinking about genomics, that one technology can do it all. Partial whole genome sequencing, Les and I have had an email exchange before. I agree with them. It's really hard to call it. We're not quite at the point of whole genome. We're near whole genome. We're near exome, but we aren't complete yet. And in those spaces are very interesting things. But we have to think really hard about what technologies we want to address very particular questions. And so the world of just running these sequencing technologies cannot necessarily reveal truth in all forms. And I think that that's an unfortunate misconception. It sounds trivial to say that, but we continue to see that over and over, and certainly reviewing papers and seeing things come through where people are misunderstanding the context of the questions that they can or can't ask. So I want to sort of focus on that. And then a single study is very good, but it does not necessarily tell the whole study unless it was a study of the entire human population, and then no longer is that a study, but then that's really anthropology. And we're not talking about anthropology today. We also know that significance has really sort of shifted as technologies have come along. We see the bar continues to change as we have better technologies, we have more individuals. We sort of continually are pushing the limits and thinking about what is it that we would consider to be publishably significant per se. And I think we have to recognize that there's a dynamic with both our technologies and, more importantly, our analytic strategies that we continue to evolve with that. So the bar continues to change. In some fields it moves up, some fields it moves down, and I think we have to think really hard about that, because that gets at the issue of reproducibility as defined by what's the coin of the realm for us, and those are papers. Those are peer-reviewed papers that are out there. Now, I think, we have, courtesy of the NHGRI, very important milestone sort of marker structures that are out there, from the Genome Project to the HapMap, to the 1,000 genomes, and these are critical tools, but by themselves, they're not studies necessarily. I mean, we conduct studies using them and going into them. And I think, we have to ask this question, do more technologies, and larger studies accelerate discovery? I think yes, but not always. And so, again, this is meant to really think about what is the context of what we're doing. So I want to sort of lay out in thinking about this, what I think are sort of the basic principles of discovery in genetics. One is a stable study design, thinking what question are you asking? What population are you looking at? How have you ascertained? Who is it that you're looking at? What question? Are you looking at common variants, rare variants? Are you looking at outcomes related to therapy? Are you looking at etiologic questions, environmental interactions? These are questions that are critical in thinking about how reproducible are going to be the results that you're going to look at. What's that sample handling look like? And I'll unfortunately make some confessions in a minute in running a large genotyping facility of errors, and how in the research setting, unlike I think in the clinical setting with CLIA really drives this notion of the chain of custody, in the research setting we are not, as much as we'd like to, nearly as careful as we are in the clinical setting. What is the accuracy of that technology? That's a very important question. What are the appropriate statistical tests that we're thinking of? What's the confirmation of that signal? Is using another bioinformatic tool good enough to say that that's indeed true? And this is a big argument. You have to go to a second technology. In the world of GWAS, we did do that, and I'll show some examples of that. And I think in next generation sequencing, that's still very much on the table. Can you look at it in different ways and say that you've confirmed it? Or do you really need to go run that sample with a different chemistry or a different technology, or run it on the same machine at a different time to be able to see it? And there are many opinions, but I think you have to at least address that question, thinking about the veracity and the strength of what your study is. How is it going to replicate? Is it going to replicate in your hands, or in your competitors' hands, or in something that you don't know about? And I think replication is a central part of this. And then what is the measured interpretation? And we'll certainly come back to that towards the end. And most importantly, what's the availability of the data? Because in this day and age, as I think Eric referred to, the democratization of this information, it becomes very important for others to be able to look at and ask the question, what is it that you've actually seen, and on what grounds have you taken that primary information, particularly from the genomic technologies, and been able to make sense of that? So these are sort of, I hope, are some of the basic principles that we'll continue to talk about, and I'll address each of these. So for study designs, we always have this question of, are we lumpers or splitters? Are we willing to sort of live with things being a little bit different, and we like the Venn diagram to say we're looking at all people with prostate cancer, even though we know there are different types of prostate cancer? Or, these are the kinds of questions that are very important in looking at the reproducibility of findings, because sometimes those decisions can be too lax and we can get in trouble. What are the appropriate comparison groups? We certainly have seen some notable papers where people have published having very, very questionable control groups or comparisons, and then when you look at the real scientific question that's put forward, you have to ask the question, was that the right choice? Is there sufficient size for discovery based on the power estimate of what question you're asking? What's your selection of the non-affected, going back to those comparison groups? Particularly, I think, in the world of next-generation sequencing, this is very important. But the other issue is really, what are the known covariates? As Dave really referred to, those uncontrollable things in the environment, they're not completely uncontrollable. I mean, so if you're going to study lung cancer, you ask the question, are you looking at smokers or non-smokers? How much did they smoke? And these are the kinds of things that we can't ignore, because indeed, for really reproducibility, we have to try and have a context for the application of the genomic technology to what our understanding is, the specific questions that we're trying to address. So sample handling. We know that there are errors all the time in laboratories. I can tell you we've run a large genotyping facility. We've genotyped almost 500,000 individuals, about a third of them or a quarter of them in studies where they're unisex, either for prostate cancer, ovarian, breast cancer. And I'm embarrassed to say, but we see about 0.5 to 1% gender discrepancy when we do the STRs or the SNPs before we put them on the chips. And we've been able to go in some settings and see that some cohorts, when they go recruit people, they'll get husband and wife, and there'll be a mix-up of the tubes, because you can go work that out. We got to understand that there are errors that take place. In the world of GWAS, this gets washed away with the large numbers and the kinds of testing approaches we have. But there is a dirtiness to this, and this also happens at the single sample setting and family structure. Those who have done linkage certainly can find that this does not make sense. So I think how we handle our samples is very important in thinking about very concretely what the laboratory approach is towards the chain of custody, from the collection to the biorepository with LIMS, the genetic barcoding and testing that. We don't put anything on without an STR. And like I said, we often, have 0.5 to 1% where we say, sex discrepancy for a unisex study. Those samples are going to be removed and not necessarily analyzed in that particular study. Management of data flow, and then, of course, the QC metrics. But I think sample handling is something that we take for granted, but we shouldn't in this discussion of reproducibility. So when we look at the search for common variants in complex disease, we know we have a highly reproducible, a very gentle technology. Illumina and Affymetrix have these chips that have 99.6, 99.7% reproducibility. If you take the same samples and you run them over and over on those chips for single nucleotide polymorphisms. And it does create a bit of a lion for us, and that is the multiple testing problem, the hard challenge of how you put all this data together. So as we know from the SNP microarray, we can actually genotype, and now using 1,000 genomes, you can impute, so you can look at those things that you think are there with very high probability, where we know there's very high concordance. But nonetheless, it does tell you that you have markers, but you have to go figure out what do those markers actually mean. So there's a strong commitment to mapping. So again, this idea that one study is going to get you to the answer is a fallacious idea. It's a series of studies. So you get this tidal wave of data, and you have no idea of really what to do, or at least we didn't. But I think, as we thought about addressing the multiple testing problem maybe 8-10 years ago, as GWAS was starting to come on on the market, so to speak, we could see that there were these type 1 and type 2 errors, and the decision was to go towards, really favoring trying to minimize the type 1 errors, and so setting a very high bar of what we call genome-wide significance. And so this is something that's been set by the community for now 7 or 8 years, and it protects against those false positive findings with a high probability that marker is real. But at the same time, we're losing an awful lot, because underneath that, because of the complexity of the heterogeneity and sample sizes, there are many signals that are being missed in any given study. And as we try and agglomerate and put them all together, we see some of them, but we really don't. We clearly are missing a large fraction of what we think are part of this sort of polygenic model, where indeed there are probably hundreds of variants that are contributing to most of the complex diseases. And we don't necessarily have the resources or the capability to put together 500,000 cases of breast cancer or 500,000 cases of prostate cancer that we see statistically will work that out. But we are slowly moving in that direction, and I think with imputation and larger data sets, there are some very clever discovery tools that are being used to look at these polygenic models and try and pull out more of those individual variants. But then, again, it again raises this question of how important is the overall question or each of the individual markers? And again, that's where you have to think about what questions you're asking, and that's where reproducibility is important for the question that you're asking, not the one that by chance or just by dint of your design you're able to at least sort of glance at, but you have to be careful not to overinterpret that. So going back to about 2007, a number of us actually in this room were part of a long discussion about GWAS, and we came up with what we thought were points to consider. So things that would need to be publicly available and be considered by the authors as they move towards this, the study information, data availability, quality control metrics. And I think out of this, we really can see some 32, 3,300 regions that have been identified in GWAS and published in the literature, that almost all of them will meet the metrics that have been laid out as sort of the confidence intervals around which we know that these are very important steps, and particularly replication studies. But it also was a time to think about what are our reviewers going to look at, the strength of the observations and the analytic approaches and the statistical significance. These are harder things that the authors have to try and predict, and it really gets to how good is your data and how much do you rip your own data apart before you show it to others. And I think the GWAS world has really set a set of standards and bars that really do force the investigator to have to try and meet these kinds of metrics. But the hardest part has been the careful interpretation of the results. From having written many of these papers and reviewed and read even more, the palaver of most of the discussions go on and on in things that are really somewhat distant from what's been found, the quote-unquote, the biologic interpretation. So we also know that we have to confirm the second signals as we move from Illumina, for instance, to TACMAN. And this checking of the concordance is very important because in our experience doing lots of GWAS, we've actually probably not published 10 or 12% of what we thought was genome-wide significance, particularly with imputation in this day and age, because that second technology does not bear what we see. So this agnostic indirect testing really begs this question of, do numbers really matter? Because when we're too small, we have a much higher false discovery rate than we would like. And when they're too large, can they really be too large? No, because it helps us to get a more precise estimate of the effect as well as the statistical significance. We know that replication is important not only in your own hands, but in your competitors' hands. GWAS has really, I think, really pushed the limits on seeing competitors as really colleagues, per se, in publishing separately and then as well as together in large meta-analyses and going into different populations and thinking about what we've sort of dubbed the cone of truth, where we go through a sequence of studies and get to these points where we clearly know that on multiple studies we can see the same signal. So here's really, in my mind, the key slide, the interpretation, where correlation does not necessarily imply causation. We know that in the world of GWAS and linkage, we've always had these indirect approaches where we have been focused on getting to a certain point of finding markers that we then have to go under the hood to identify. And I think that that's really where we've been very successful in genetics. I think it's harder when we look at NGS and we're tempted to think that we can go right to causality by looking at these things. Our signal-to-noise ratios are still so blurred that it's very, very difficult, with rare exception, to be able to pull out those variants out of exomes and say, aha, this is the explanation. I'm going to ignore the 5,000 other novel variants that I've seen and just pick 5,001. To me, there's great danger, and this is where replication and reproducibility become absolutely essential. And the last point about this, I think, is the DB gap question, the sharing. I think the whole issue of the genomic data sharing policy has been a critical step towards pushing people towards really understanding how you can use this de-identified information to, most importantly, do independent analyses to confirm. And the reproducibility comes not only in the hands of the investigator, but the community to be able to look at this. And we clearly have some examples of this where this has been a real issue. So sort of heading towards the end here, I want to talk about the balance of genetics, where we really have balancing between statistical significance and the efficiency of those tools. And the money is going to push us one way or the other. And this is a challenge that, again, going back to the hierarchy of how you get to those thresholds, we are continually challenged in terms of thinking, gee, we'd like to do more individuals, we'd like to do more technology. But really, in the discovery, there is this balance, and we have to recognize what is the utility of really what we're going to find. So let me I want to skip, I took a little bit longer than I wanted, and I'm just going to go to the last slide, the last two slides here if I can. Okay, yeah, here, that's it, okay. So I want to finish by saying here, I think we really have to be careful about our genome analysis, which is both promising, haunting, and at times very dangerous. And as we go across the spectrum, we really have to think about the final words are know what you're looking for, what your models, your types of variants, and the components of the genetic variation that you're looking at. And also know what you're not looking at. And this is something that we have to be more honest about. We do know that we can find some things unexpectedly if we open our eyes to, as we did with our GWAS and doing 100,000 individuals, finding that there was this detectable mosaicism. But again, the scope of discovery is very important, and this will help, I think, to limit the issues of reproducibility. So why don't I stop there and turn it over to Les. Thank you.
Okay, great, thanks. We'll just launch into this with no further ado. So the issues at play here are overlapping with what Stephen was talking about. Drilling down a little bit more, one of the things that our field is currently struggling with in genomics are two levels of assertions that people make, scientists make, which are gene-level assertions and variant-level assertions. There are issues at both levels. I would suggest that the second one is a much larger problem than is the first one if you talk about the scale of issues that we're dealing with here. But we argue about these things, and sometimes these arguments can get a little ugly, and those of you who are old enough will know what this argument is like. And sometimes they get a little unseemly, and we're going to try and move this towards a more civilized approach to the argument and talk really about this issue openly. And we have to acknowledge that we're not hanging crepe here. It is not as though the sky is falling and you should ignore all published genomic studies because we have so many problems. That is absolutely not the take-home message. These studies work, they are powerful, and we have to know when they're giving us the answer, we want the answer that is valid and true and reproducible, and when we need to be more skeptical and how we can move through to get the glass more than half full. And so what I want to do to try and review a few of these things is talk about some of the technology and discuss the approaches. So Stephen mentioned sort of the old school, what we were doing 10 to 15 years ago, which was this positional cloning paradigm. And through that gauzy sepia tone look in the past, it all seemed beautiful and simple in the past. And it wasn't quite as simple and lovely as we all thought. In fact, there were some real flaws with that. One of my favorites is what we used to do, remember, when we did positional cloning, is we would do this drill, find variants, and then we would go back and ask the question, how commonly do I see my reportedly pathogenic variants in a control population? That was a completely bogus statistical approach to understanding controls, because what we now know, because we have the genomic tool, is you want to know what the frequency is of variants in a control population, not just the ones you happen to find in your cases. Right? And we couldn't do that because you couldn't sequence hundreds of DNAs using Sanger technology, so we all conveniently ignored that little problem and swept it under the rug. It was a problem. All right, but NextGen has swept away all the intermediate steps between a proband, a proband and a variant. All right? This is wonderful. Right? This the democratization that Eric was talking about, This is faster, it is cheaper, lots of people can do it. We don't need these annoying linkage and statistical people hanging around our labs all the time. We don't need to make libraries to find genes because they're on a web page. We don't need all these experts. We just send the tube of blood off and we get back the answer. It just falls out of the sky. Isn't that great? It also has practical advantages of things like we don't have to have gigantic Mendelian families to do these. We can do single cases. We can do de novo. We can do mosaic. Okay, that's all the glass half full. And this is also the glass is half empty, because all of these things have created a whole set of challenges and problems that we now have to deal with that are different from the problems we had to deal with before, and so we have to figure out how to address them. Okay, so, but I think the crux of the problem is sort of the search space false positive error. Steve showed the nice 2x2 matrix here, and what positional cloning did for us is it massively constrained our variant finding problem by constraining our search space from 3 gigabases of DNA down to a couple of megabases. That was a great thing, and it reduced type 1 error, positional cloning did, by reducing that search space. And so we didn't come across lots of variants by chance. Now what we've done with NextGen is flipped the search space back to the whole 3 gigabases. And so now we have reduced type 2 error, that is we've failed to find things less often, but we've increased type 1. And so what does that do to us? Now scientists are not dichotomous rational decision-making machines. They are in fact humans, and humans have cognitive errors. There are things like temptation. When there's something in front of you that looks really appealing and appetizing, what do humans do? They find a way to make that thing that appeals to them make perfect sense. And my friend David Goldstein likens this to a Kipling Just So story, this elaborate argument, and we are all guilty of this. I don't know if anyone in here hasn't had the situation where they found a variant and they can immediately create a beautiful, logical, internally consistent story for how that variant is associated with that phenotype, irrespective of whether or not it's true. Right? That's what human beings do, and we have to recognize that that's what we do and build in systematic, objective approaches to addressing that and eliminating that error that we tend to make. Now, as Steve said, our GWAS colleagues have solved this problem. They've solved this problem in an objective, rational way using a single statistical model that objectively addresses a unimodal data type, right? The beauty of GWAS is it generates only one kind of data, and you only need to do one thing to understand whether it is statistically valid or not. And so you do this, and this is beautiful. It's simple, it's clear. But for every complex problem, there is an answer that is clear, simple, and wrong. And the problem is we now have in NextGen is that a p-value of a variant is not biology. And what we need to be doing is, as Stephen alluded to earlier, and David as well, is think about this in a much higher dimensional sense, that there are a lot of things at play here, and that when we're doing this process from start to finish of establishing causality of both genes and variants, we're using multiple kinds of data, heterogeneous types of data, simultaneously, each of which has error modes. The genetics is sometimes the least of our problems. Often it's the model systems and the functional assays that are much harder to analyze objectively and statistically. And so our problem is one of dimensionality, and this is a tough nut to crack. Now, our institute took a crack at this a few years back. Some of you were at the meeting at the Bethesda Hyatt where this was discussed. And to try and tackle this, what are these dimensions of data that we're trying to incorporate and merge and rationalize in order to get from variation to causality? And this paper laid out some of those key considerations. And it, like the weekend update, led to, let me say, a bit of controversy. So there was a response from a very well-respected immunogeneticist who was fairly unhappy with the Nature paper and had a basically a response paper in JXMed that laid out criteria that he felt were completely opposite. The Nature paper went into a number of considerations of how you design larger, more powerful replicating studies. And Jean Laurent says, wait a minute, we can do this with one patient. Here's how we can do that. And I think actually this is a good example. Here's a summary of Jean Laurent's causality criteria, which might in fact be a special case, and I think there may be some validity to his argument. And I think some of these arguments are people sitting in a room and saying apples are red, and then the other person says to them, no, you idiot, oranges are orange. Right? And it may be that there's truth to both sides of this, and Jean Laurent has a particular scenario where a single case and a single exome and an appropriate functional assay can get you from variant to both gene and variant causality in a robust and reproducible way, but that may not be generalizable. So my conclusion from this part of the talk is that the GWAS approach, this unimodal statistical approach, single-dimensional statistical approach, worked for GWAS, but I do not think it is generalizable to our current problem because it is highly dimensional. And this is a tough one, is integrating data of multiple types, and every study is a slightly different combination of these different data types. And so the Casanova criteria work for a subset of scenarios. They won't work for most. And what we have to do is figure out a larger system for analyzing such complex and dimensional data going forward. Now, some groups have started to tackle this. The ACMG, in concert with our institute, has set up working groups to try and crack this nut, both a clinical and research sense. And a paper that recently came out from the college as far as how do you go from variant to diagnosis, so this is more of a clinical sense, is this matrix. And this, don't need to read this. I'll give you an example. It's a whole set of different kinds of data, this dimensionality of the problem we're facing, what different kinds of evidence of those different types might look like, and how you can evaluate that. Just give you an example. Here's one supporting criterion for pathogenicity, which is you have a missense variant in a gene for which there is a low rate of missense variant in control populations, and that in diseased patients, pathogenic variants are common. So there's a semi-objective criterion, but if you start to think about that sentence, take a step back from it, there's a couple little, I call them weasel words in there, right? What does low mean? What does benign mean? And what does it mean to say that it's common in patients with the disease? All these are actually semi-subjective terms, and we have to work through data in order to understand how to quantitate that and objectify this term, even though this is certainly progress. So then you take all these different data type attributes, and then you merge them, and this is all color-coded to make it a little easier, in different combinations. And this can be regarded as an early first attempt at this high-dimensional data problem to integrate them in order to get to an end determination of something between pathogenic and benign. And so this is an attempt on one scale to do this. Another attempt was incorporated into the Banbury conference, which occurred last year, which generated 3 challenges of this process. First, again, is to really nail unambiguously this gene-disease relationship phenomenon. And again, as I said earlier, I think that's a little bit easier of a problem than the variant association issue. But to, again, move towards objective criteria for establishing the pathogenicity of individual variants, and then figuring out how it's not enough to just say that this variant is pathogenic. That's sort of a pre-hoc single assessment of a variant. What we actually are doing here is doing a Bayesian analysis of taking into account an assessment of a variant and coupling that with what we know about the question that was asked that led to the data being generated in the first place. That's the design issue that Steve was talking about. And then doing a Bayesian analysis, which is currently mostly done in an ad hoc, informal fashion. But I think we need to formalize and structure that and actually do that in a quantitative way. And again, that's about differentiating pathogenicity from solving the problem that you're being faced with, which those are two different things. And how do we move from the general problem we have in science to N of 1 cases? The N of 1 problem was exemplified by this paper, which appeared very recently, which is talking about one-person trials. Now, this is the very antithesis of the Fisherian frequentist approach to statistics, which is huge studies powered pre-hoc to answer questions, followed by a comparison of various Fisherian statistical models. What's being asked here is a different question, and they're pushing us in a direction that I think is interesting, challenging, and we're going to have to work hard on, which is to think of this in a more Bayesian context and move towards answering questions in novel ways. And Bayesian approaches can be just as valid as Fisherian methods, and genetics has a long history of doing this, and I think that we can begin to explore that space and address these questions. So, next-gen sequencing, gene finding is certainly complex. Type 1 error is a huge potential issue. I don't think that a simple unimodal statistical approach is going to get us where we need to go. And I don't think that we can only consider frequentism and Fisherian statistical approaches and demanding huge RCT, randomized controlled trials, for every question we want to ask. It's not going to work going forward. And we have to figure out novel ways to answer these questions because, again, of the dimensionality of the problem. Glass half full. Frameworks are starting to be developed where we can, at least in a semi-objective manner, start to understand our evidence and begin to quantitate it. Currently, these are all heuristic approaches and require judgment and subjectivity, and we need to shift that over time to more data-driven, objective, and hopefully more automated approaches, and to develop new study designs. So in closing, I'll just say this is a hard problem because of its dimensionality, but it's really important not to lose sight of the fact that it overall works very, very well. And so we don't need to be hanging crepe about this or apologizing for our science. We need to improve it for sure and develop new methodologies and keep moving forward. I don't actually think that we will ever get to the point where we'll be able to put blood into a machine and an answer will come out of it and we don't have to think about it. I don't think we're ever going to get there. Human judgment is always going to be needed at the end of that process to make it make sense. And one thing is for sure, this is the most exciting time you could ever possibly be a human geneticist because of these tools, because of the power, and because of the challenges. And so I've enjoyed it tremendously, and I think we're going to have a lot of fun in the next decade or so solving these challenges. So I will stop there, and then we can take questions.
Yeah, thanks. So part of the reason that we had journal editors here to help moderate these sessions was because there are roles for journals in maintaining reproducibility in a field. And I just put up a few thoughts here before we open the floor to questions. So, you know, journals don't really set standards. They follow the field's lead and hear from the field what those standards are. But we do have a role of sort of enforcing them. We publish guidelines where necessary and appropriate, and you saw a couple of examples of those from Nature and Nature Genetics and GWAS and in variant interpretation. Journals, I think, should be open to confirmation and alteration of authors' work through companion articles, through letters, through corrections, additional data, and, of course, retractions. We'd like to be consistent and offer guidance to authors up front. And of course, we need to be open to advice from reviewers, from editorial boards, from the community in general. And AJHG, as a society journal, benefits from many inputs for those areas. But we also maintain standards, and keep in mind, as we saw, the bar always rises. The standards are always going up. And the journal needs to provide examples of what the current state of the field is. but also provide a forum for authors to make assertions based on their best evidence and the latest techniques and currently accepted interpretations, and not be afraid of those assertions being wrong. And I think my bottom line, particularly for our journal, is we are often publishing clinically relevant data, and we want to minimize delays to that publication as best as we can, because it's important to people who are have patients with similar disorders or similar mutations. So with that, and I know Hillary has some remarks about genome research in the next section, we'll entertain questions about publications, about interpretations of variants. Who has a question?
Well, I'll take Jane Curtin's position to one of the things you said last, the point and counterpoint.
Yeah.
I think, it gets to the N of one, and the Casanova paper is as attractive as it is at the very edge of the spectrum of having very remarkable circumstances. I think we as a community have to be very careful in embracing that. I mean, if I look back, and think of when I graduated from medical school, I took the Hippocratic Oath, I would do no harm. And I'm worried that the insufficient and incomplete use of genomics can do an awful lot of harm. And we see this in the breast cancer world, where women with BRIP and BARD1 mutations that have effect sizes of 2.5 and 3 are being recommended to have the same body-altering surgery that the BRCA1 and 2 mutations have. And so the associative property is a very dangerous one in the hands of the physician. So I do think that as we look at this N of one, we have to be extremely careful in when, where, and how we would make that transition from discovering something to actually clinically doing things. Because clinical genetics is not only making the case, but there's a recommendation. There's something that's going to happen on the basis of that. So I have a much saltier and a much more, difficult view of the N of one. I mean, I realize in cancer we do it for exceptional circumstances, but I do think we have to be very, very, very cautious, because I think we really do need the reproducibility, and we need the other confirmatory studies before we start making very important recommendations.
I would just say everything you said is true, and I disagree with you. And I think, of course, I think that Jean-Laurent picked some sort of almost cooked examples to make a case. And however, that being true, when you said the Hippocratic Oath, which is a ancient principle, I actually have a slightly different view of that, which is I say to people, you have to tell me honestly, what mistake do you want to make? That's what you have to tell me. And I think the answer to that question tells me how I should design my study, how I should power it, and how I want to trade type 1 for type 2 error. And there are some cases where I'm willing to tolerate type 1 more than type 2, and the contrary can also be true. So I don't think we can take a global view of saying do no harm, because it actually denies what I see as the reality of clinical medicine, which that it is frequently and uncommonly pathetic what we do to people in the clinic. We make decisions all the time on really crummy data, huge amounts of crummy data, human error, decision-making errors all the time. And you can say, and I agree with you 100% about the imperfection of genomic data. And then I look and I see what data people are making decisions on today, and I'm saying, I'm not that sure the genomic data, as imperfect as it can be, is worse than what we're doing today. And that's not to say that's not an excuse for crummy science. It's not an excuse for lowering the p-value from 0.05 to 0.1. That's not what we're about. But we have to say, what is this going to be used for? How do we make the decision? What's the validity of the data? What are our error modes? And which errors do we want to tolerate? And how do we go forward? So I think it's actually an interesting, very nuanced problem that has different answers in different scenarios.
I don't disagree with the last thing that you said particularly, but I think that it does tell us that empiricism will come about with the collective, archiving of this information. And I think the danger is up front making these kinds of decisions and making these interpretations with relatively little data and acting on that. And so I think that's where the question really, in my mind, maps to how we capture the information. So at the fifth or sixth patient who has, an LOF mutation in gene Q is really very interesting. The first and second is part of the discovery in my mind, and we have to be very cautious in going forward with toxic therapies and all sorts of remarkable, interventions. And I think there's that transition period that we have to recognize that we're in, and that's why I'm a little more skeptical about, going so quickly to the clinic with some of these things. And that's, that's more the oncologist in me, I guess.
And, Joan Bailey-Wilson, NHGRI. I agree with both of you because especially it really comes down to what error can you tolerate and what is the purpose of what you're doing. So I think you're both right, and it's really complicated. But one thing I will say is, Les said, we don't need those linkage geeks anymore, but actually,
we do.
This is where those linkage geeks can really help you. Because if you have one patient in a family, like that nice dominant pedigree that Les showed you, what most people then ask is, we've got the variant in one person. And you say, does it segregate with the disease in the family? Guys, you're doing linkage. You're doing linkage under a very small set of assumptions that says it's 100% penetrant and there's no phenocopies, and that's one type of linkage. Once you get from the simple case, though, into more complicated diseases where maybe you've got some phenocopies or more likely you have some gene carriers who aren't affected, this is where the linkage geeks can run linkage accounting for that and really help you verify that, yes, that variant does segregate with the disease as you'd expect in this family. So don't write us off. We're there to help.
No, of course I agree with you, and I'm a person, I can claim to be a linkage geek. That's why I've run FastLink from the command line, so I can do that. And however, with the nuance, so I agree with everything you said, but the nuance there is when we're doing this in NextGen, where we're starting from a variant in a proband, primarily what we're doing with linkage there is a gene phenotype association. It actually, it's not a variant disease association because of the Steve slide, because of the LD issue, right? We have to be very careful about that. Yeah.
Because I think that's a key issue when you come back to the design and particularly the interpretation, and that is less really, I think, characterized it very nicely, the gene versus the variant. And GWAS, a lot of it has been about variant. And if we look at TCGA and the ICGC with respect to cancer, one of the really striking features is many of the things that are important drivers are not necessarily in the gene per se. They are rearrangements, they're regulatory regions, things that are disrupted. So the idea that we would understand is a second order activity, the disruption of a particular pathway or the expression or shutting off of a gene or the creation of a new, a chimeric protein or whatever, does stress this issue of thinking at the gene level only. And I think, again, that's where we come back to the reproducibility is such an essential part of this science, and seeing either in additional subjects or studies that the investigator may conduct, or in others who independently do this. And this sort of gets at a delicate issue that I hope our editors will talk about, and that is the novelty of reproducibility, which is a contradiction in terms to many editors, but I think as we look at genetics is really a very important question. Does somebody see the same thing in a completely independent study? And to us, I think, as we particularly think of the clinical thing in the Casanova example, that becomes all the more important, because if we don't agglomerate all and put together all of these cases, circumstances, N of ones, we're never going to solve that N of one, much less in the discovery of where we think there are important genes or variants. It's really these independent, separate replications that really give us the I think, the strong confidence that this is an important finding worthy of the next generation of studies and or implementation.
Michael?
Just to pick up on one of the comments that Steve just made about independent verification, one of the themes that's run through these workshops that we've been having is the need for independent verification using another approach. So, for example, in the field of structural biology, although the crystallographers held sway for a long time, people are beginning to sometimes question crystal structures and say, well, what does NMR show? What does cryo-EM show? Are we seeing the same structures? For high-resolution optical microscopy, what's the EM show? Does it correlate with what we're seeing? So is there an equivalent in genomics? And one of the things that we sometimes ask of our scientists is if you make a model system, for example, can you replicate the findings? As we get more sophisticated in phenotyping patients, can we see actually a spectrum of phenotype which is different from the wild type in various kinds of variants? So could you comment on other technologies other than genomics to verify genomic findings?
I will. I think when you go into that world of between the Mendelian disorders and the GWAS world, those moderate penetrance, moderate effects, the only ones that have been established with correlative laboratory animal models, cell lines and things where people can say, gee, the MITF simulation pattern in melanomas is different here, and that makes sense, and it correlates with the statistical significance that we see that is not quite at the bar that we would like to see with the less common variants as a power outage. And I think, a number of the journals clearly with a lot of the exome sequencing going on are requiring that you do more than just say, we declare this to be significant because we see this segregating in the families, but rather, what is the laboratory basis? Is there something that really would point to this being important for diabetes or cancer or what have you? So I think that's beginning to break out, but it isn't breaking out quite as much as we'd like. I mean, I'm sure the journal editors see lots of papers that are coming in purely as genetic marker studies for that whole space, and that, it's an important tension where the field is. Now, if the frequentists win and we have 100 million people sequenced, then we may be able to start looking these things up. But we know that that's certainly a long way down the road. What? And completely phenotyped, exactly. Yeah. But there's no question that the causality has to have more than just the marker identified, with rare exception. Now, the Casanova arguments still has the causality established by correlative studies, functional studies.
Right, and that's their huge advantage. Is at that meeting for the Nature paper, we talked about the proximity of the functional assay to the disease, and I think that's the advantage that the immunologists have, is the things that they can do in the flow sorter and in the dish are very close to what's wrong with the patient. The opposite of that is what you mentioned, David, are the behavioral studies. And so when somebody says, I have an intellectual disability gene and this subcellular compartment looks abnormal in my functional in vitro, we got some distance there between what's wrong with the patient and what that functional assay is showing. And we can understand that in a general way, but actually quantitating that concept, that's a really hard thing to do. How close is it to what we really want to assay? And we have to work a lot on that.
And we'll take one last question. Thanks.
So just real quick, what do you think of sort of the ACMG sort of categories that are used for classification of variants? I know it's kind of like throwing a meat chunk at a pack of wolves or something, but just curious, should it stay 5? Should it go to 2? Should it go to 8? I'm just curious what your thoughts are.
So there's two kinds of people in this world: those that divide things into categories and those that don't. And so it's human nature to try and categorize things. And then as soon as you do that, you have the ones just above the cutoff and just below the cutoff, and everyone agrees it's bogus to differentiate them. Now, there's actually been pretty good behavioral data, and we need to go back to that, to look at when human beings says something is highly likely, what that actually means to people numerically. And there's really cool studies that have done that. And I think the 5-category approach with 2 very thin slices at the end, 2 larger ones coming in, and then a huge middle of vagueness, actually pretty closely approximates how human beings really fundamentally think of risk statistics. That's not to say that those 5 are exactly right, but it's pretty darn close to that. And we and others are doing experiments to see if we categorize variance this way or that way, does it lead to differences in how clinicians and patients view things. So it's a really good question.
I mean, I would just add that I think that the categorization at the ACMG is the great tail to wag the dog on the incidental finding question, because I think one of the hard things is, you know, as I had in one of my slides, know what you're looking for, be open to the things that you may find as you go along, and as we do these exomes, you may be looking for a diabetes family, you know, for something, and then all of a sudden you see there's a BRCA1 mutation. And so I think we do need guidelines, and to be sure that when we transition from what we've looked for to what we've incidentally found, it's very useful for that. For, I think, all the specialists who are interested in those particular genes, it is very, very helpful, but it's a dynamic. You know, the clinician wants one of 2 things. Do I act on this or do I not act on this? You know, the somewhat probable, the VUS, all those things, you know, do I need to tell the patient they should do something different or not? That, you know, it's a dichotomous question at the end in the clinic.
Okay, with that, I think we'll end the first session. Michael, are you going to introduce the next one, or shall I? Okay, our next, thank you, gentlemen. So the next session, I think, turns to more technical aspects of sequencing. And Hilary Sussman from Genome Research is our moderator.
Good afternoon. I'd like to thank Stephen Chanak, Michael Gottesman, and Paul Liu for the invitation to come here. It is very helpful for me to be involved in these discussions, which often suggest improvements the journal could make and inform on policy changes to meet and raise community standards. It was clear when the Bermuda Principles were established in 1996 and with the Fort Lauderdale Agreement in 2003, I think Dr. Green hinted at this before, that genome sequence data sharing was of paramount importance in the genomics community. And with the massive increase in sequence data being generated and the ever larger scale of these analyses, data reproducibility in genomics has become a very timely topic, and one that requires an open discussion such as the one we're having today. So without further ado, I'd like to introduce the speakers of this session. First up is Dr. Jim Mulliken, who is the director of the NIH Intramural Sequencing Center and an associate investigator in the NHGRI Cancer Genetics and Comparative Genomics branch. He was involved in many aspects of the Human Genome Project, as well as numerous other whole genome sequencing, microbial sequencing, and cancer genome sequencing projects. As a computational genomicist, he develops new methods to better interpret the rapidly expanding volume of sequencing data. He will talk about the experimental considerations and biases in conducting whole genome and exome sequencing experiments. Our second speaker is Dr. James Taylor, Associate Professor in Computational Biology and Genomics at Johns Hopkins University, whose genome informatics group develops approaches to enable large-scale genomic analysis to better understand genome structure, regulation, and evolution. Most notably to me, Dr. Taylor contributed to the development of the Galaxy framework and to many other software and infrastructure, most of which are over my head, especially those in the cloud. The stated goals of his group and the topic of reviews he's recently published is to make computational biology more accessible, transparent, and reproducible. So it is fitting that he will talk about reproducibility in computational analyses today. After Dr. Taylor speaks, I'll say a few words about how genome research promotes reproducibility, and then we'll open the floor for questions.
All right, thank you, Hilary, for that introduction. And I'm just going to dive into some really technical issues. I think it's important because, as Eric Green mentioned earlier, there's a real democratization of sequencing technologies out there. And as more and more labs bring them in, I hope what I say today will help people avoid pitfalls that may befall them if they didn't know the ways that you can go wrong in this, and hopefully steer them into keeping things with current best practices. I'm not going to dive into all the different technologies that are available. There's a good review as listed at the bottom here. There's a variety of different technologies that are out there or have since been retired, but Illumina, the Life Technology Systems. I put ion torrent and ion proton in parentheses because those weren't reviewed. That came out, I guess, a little later than the review that's listed below. But what I'll be focusing on are those technologies that generate the millions to billions of short DNA sequence reads. Just as a bit of an overview of this process for genome and exome sequencing, one starts with constructing a shotgun library of a given short fragment size, 400 bases, let's say. This library that's been created for the given technology that it's going to run on is either PCR amplified. Typically that was the case in the past. There are some methods now that are PCR free, which avoid certain pitfalls that I'll describe later. Those are loaded onto the sequencing machine and they generate your millions or billions of reads that you then map, align, and call the variants for that. Looking at exome sequencing, it's very much the same process. But after you create the fragments, the library, you do typically, I think in all cases, have to amplify these before you go through the hybridization step. The hybridization step has probes that attach to the regions of the genome that you're interested in for that sample. Everything else is washed away. Those are pulled down, captured, and released. And then again, another PCR amplification step is necessary to give you enough product in order to load on the sequencing machines and again, generate your millions or billions of sequencing reads. This whole process of exome sequencing is highlighted in this review that I list below. So moving on to the next steps, aligning sequence reads to the reference genome, there are many choices for this. And this can be a source of reproducibility challenges is if one group used BWA and another group uses NovoLine, how well do these agree in the end? There are differences in the ways these different software packages work. Again, there's a review article to help understand the different methods for aligning sequences to the reference genome. But then what is the reference genome? For the human genome, there's been, I'll show you later, many different versions of the human genome. But one particular pitfall that can befall you is there's one region of the genome that is represented in 2 copies, whereas the rest of the genome, all the autosomes are represented only once, and that's a pseudo-autosomal region. It is present on the X and the Y chromosome in the human genome, and in order to make the methods that work for the rest of the genome, the autosomes, you need to mask out the region on the Y chromosome when you build your database for alignment purposes. So that's a particular pitfall. And moving on, once you've aligned your reads, you would think, well, if you look at Poisson statistics and you generate 5-fold coverage of the genome, you should have most of the genome covered with a read, right? But if you look and say, well, I have 5 A's here, that must be a homozygous variant. The dots and commas all agree with the sequence that you see here, and these 5 A's say, hey, maybe it's a homozygous. Well, I say maybe because I know that in this particular case, if you look at enough examples, it's really a heterozygous position. So this is flipping a coin. For the heterozygous positions in the genome, you can, if you do it 1 million times, because, well, let's say there's 2.5 million heterozygous positions in the genome, flip a coin 2.5 million times, you will see things happen 5 times, 10 times in a row. So you need to overcome that with the power of statistics. And with the next-gen sequencing technology, you can get to the depth required to do this. So once you get sufficient depth of coverage of your genome with these short sequencing reads, there are methods that are mostly Bayesian-based, BAM to MPG, GATK, SAMTool, SOAP, SNP, and FreeBayes, to name a few, are some packages that you can use to call the genotypes across the genome. Typically, you'd need about 20-fold read depth to get proper statistics for a whole genome. So when you're doing whole genome, aim for at least 30-fold coverage or higher, and 50-fold or higher coverage for exome sequencing is a standard practice. So I've hit these first 3 topics here, sequencing, alignment, genotype calling, and mentioned some of the pitfalls. I'm going to continue on through these next 5 topics here that lead to pitfalls to reproducibility. The experimental step, one of the first steps of just, are you starting from a cell line? When cell lines are immortalized, it creates genomic alteration. So that is a change to the genome itself. And how many cell line passages has this cell line been through? That can result in differences in the results if you started from the cell line at an early stage of passage versus a later stage. Oftentimes, especially in cancer sequencing, you may have limited amounts of DNA, and you need a minimum amount to really get started with the next-gen sequencing process. So you may need to do something like whole genome amplification. That will introduce its own variability of the genome, but necessary in order to get the process to work. DNA fragmentation and size selection. What size short fragments did you start with? And that will lead to differences in how the reads get aligned back to the genome. So if you're doing a study and let's say you're comparing tumor and normal, a sample from the same individual, it'd be best to try and keep all of these steps as consistent as possible. Finally, the sequencing library construction, I mentioned PCR amplified, and I'm going to show a specific example of where PCR duplicates can lead you astray. And this is an extreme example. I had to find a library that we had created that we rejected in the end. It was a failed library because it didn't have enough complexity to get us the coverage that we needed. And what you see here is reads aligned. This is an IGV view of the genome and how the reads align. Gray means it all agrees with the reference. The different colors, here it's just red, that's a particular base. There's a homozygous change here in this individual at that position. And these are mate pairs that are aligned. And this, you see this sharp edge. All 8 reads here start at the same position. And at the other end of this mate pair, end at the same position. These are PCR duplicates. When you amplify the sequence, you create more copies just so you can load it on the machine. But that means you can sequence that library to whatever depth and be led astray by this. And this must have happened, this particular PCR amplification error must have happened before the capture, and then it was post-capture amplified and sequenced, because in this same sample that was sequenced, these are the same samples. There's no evidence of this T allele at this position in the redone sample. So this is a particular pitfall that people need to avoid, is to know the complexity of their libraries, how many unique molecules are there in the library, how deep can you go, and remove the duplicates if there were PCR steps involved. So what are the effects? I took this particular sample since I had both the one that we failed and the one that was not failed and passed. And without removing the PCR duplicates, there were 1,500 variant call differences between this data set and the repeated exome data set on the same sample. And this is about 4 to 5 times higher than what you would see in the disagreement rate between 2 properly repeated exome data sets. Continuing on the theme of exome enrichment, there are many different vendors and versions of capture enrichment systems for exome sequencing. At the Sequencing Center, we've used 4 different ones that you can see listed here. There are many others, and all attempt to capture as much of the genome as possible. And what I want to show in this slide is what these 4 different kits look like. These are the design probes in the top 4 tracks. Black means it was targeted for capture, and the bottom 4 tracks in the same order are the actual results from a real sample. And in this first kit that came out, this was a very early version of SureSelect, and this gene wasn't properly annotated at the time, or they didn't pick up this particular gene, so they didn't design for that gene. It was completely missing. Some nonspecific binding of other probes did land a few reads in this region. But there's a variability. If you want to reproduce something that was captured with this kit today, they may not sell this kit anymore. But you probably don't want to reproduce something where you're missing data. And hopefully that as kits progress, we'll get better and better results. But here's an example. The TrueSeq kit happens to like to capture this particular pseudogene, and others didn't decide to choose that one. So each group has their own selection of genes. Again, most of the exome is there, but it's never the whole exome. So switching on to looking at changes in sequencing technologies and modalities. And another thing that will change whether you can reproduce a particular experiment is whether that version of machine still exists even. There's always new sequencing platforms coming along. You'll get upgrades to sequencing instruments. They'll change their chemistries, their substrates, their base calling algorithms, especially in new technologies that just come out. There's often a rapid-fire change of methods for producing the data out of that machine. Single-end or paired-end sequencing reads, that's typically a choice that you have for a given project, and what the read length is for these short-read technologies. Again, if you're going to be doing a particular study where you have hundreds of cases and controls, you want to make sure that you do everything with as similar a sequencing methodology as possible. But when that changes behind you and you can't control that, you need to take that into consideration, is what happens. Can you get your samples through the system fast enough that you'll have all the data out for your particular study before the technology changes? Now, this is a graph that I was surprised by when I generated it just for this talk. Here's one sample. We sequenced this one at the Sequencing Center back in 2009, and then just end of last year, and then early this year. And then another sample sequenced with different chemistry, here 2x125. These numbers kind of tell you what the read lengths are, whether it was single end or paired end, and the length of the reads. And if you look at this top track, the GC track, look at how that is kind of anti-correlated with the coverage. And this is well known by Illumina technology, but I was surprised that it was as consistent even over these 6 years that we have spanning this time frame. Now, even in light of that, it's clear that this particular homozygous deletion, the 1.4 megabase deletion, was observable at this time back in 2009 and continues to be seen. Had you not appreciated the fact that GC bias changes the level of coverage, you might think, oh, this might look like some kind of copy number change in the sample, but you need to control for that and understand that process. So, yes, sorry. So the question was this the same aliquot of DNA? And I should explain. The top sample, these were all the exact same library even. One library was run on these different conditions. Here, the 1x150 and 1x100, they were multiple runs, multiple libraries from the same sample. Yes, these are all the same sample, maybe a different extraction from the blood. And so that's an important question. So here, I just wanted to show that as we get better read lengths, yes, clearly we can align things better. This is a line element that is proving quite difficult to align to with the short single-end reads, with the paired-end 125 base reads, with a 500, 600 base insert library. We're getting some coverage. Once we get to 2x250, we have aligned across that region because it becomes unique enough in the genome that it can align in that region. The method that we use is if the read aligns equally well in more than one position in the genome, we don't align it. We don't leave it aligned to the genome. Looking at the comparison of the calls here, for the second study, this group at sample 2 at the top, 2x125, and repeating the same library on the same machine with different flow cells, we get 99% concordance across all 3 sequencing data sets, and very good concordance between the technical replicate of the 2x250 But when you look at the 2 by 125, shorter read length, different chemistry, different machine type, there's 1% difference here. And are these errors? I didn't dig into this, but these are differences. And if you want, let me go to the next slide. This might be a little bit easier to see. I've done two pairwise comparisons, each compared to this set B, 2 by 250. So we get 40,000 discordant calls at this genotype call threshold between this technology and that. And when we keep it the same type read length, the systematic errors are greatly reduced, and this is more statistical-based differences, discordant calls, because I can increase the genotype call score, and these drop down to just 52 differences in the genome with little decrease in the total bases compared. So the sensitivity decreases slightly, but the specificity is just much better. It's not saying that all these calls are right. It's saying that we're able to call the same way for the same sample. And again, very important if you're doing something like, let's say, a parental cell and stem cell comparison. You want to know which variants have been introduced into the induced stem cell lines. You want to minimize your background as much as possible. Now, just run through a couple last points here. The genome reference has been changing for many years. UCSC started with HG4, went up to HG19. Now they're calling it GRC H38 to be consistent with the NCBI genome builds. Luckily, these are all archived, so if you do want to reproduce something from somebody's data set, you can download the same genome version that they used if it was listed in their publication, and then reproduce the experiment. If you want to know more about the GRC, look up Genome Reference Consortium. Variant annotation, this, again, which gene models do you use? There's various ones to choose from. Ensemble gene models, all genes in UCSC track, so forth. What tools do you use? One of the most common ones is ANNOVAR, but the variant effect predictor is an annotation tool that is also used. And then once you know the amino acid mutation, you want to know the mutation consequence for a given change in an amino acid. There's polyphen-2, SIFT, I didn't list that one here. CAD is combined annotation dependent depletion. So these are methods using comparative sequencing data among other data sets to figure out how severe the mutation consequence would be, which in the end helps one understand which are the variants that are most interesting in this set that may be related to the disease that you're studying. And then other data resources is OMIM, HGMD, and NCBI's ClinVar database. And just to summarize what I've talked about today, and this time I don't want to say pitfalls. If you avoid the pitfalls and use current best practices, you can get through all of this, and really there's great power in the technology, and you can really utilize and realize that power. I want to thank Nancy Hansen from my research group and Alice Young and Jim Thomas for help with these slides, and everybody at the NIH Intramural Sequencing Center for our shared experiences in this technology. Thank you very much. So next, I want to introduce James Taylor. He's going to tell us about the analysis, reproducibility of analyses.
Excellent. Thank you, Jim, and thank you, Hillary, for the introduction. I have an ambitious number of slides, so everything's already available online at SpeakerDeck, and these are licensed under Creative Commons by attribution license, so feel free to take them and do what you will. So, as we said, I'm going to mainly be talking about the parts of the analysis issues after raw data has already been collected, right? So Jim talked a bit about the data collection issues, experimental design. These are really important. issues, and if you don't do your experimental design right, there's no point in any of the other stuff I'm going to talk about. But once you actually have data, then we start to move down this pipeline of increasingly more derived data, increasingly smaller data sets that get increasingly more scrutinized, right? And so by necessity, when we're talking about genomic data, only some summarized data, some summary statistics, maybe some inference, some hypothesis tests can actually make it into the publication. And so much of what's happened is kind of hidden, right? And so, but it can have a profound impact on your results. And so how is the data cleaned? How is the data analyzed is absolutely essential to understand. To parse this problem down even a little bit more, because I really want to contain what I'm going to talk about, we could think of two questions we could ask about published analysis. One would be, is the analysis as described correct? I'm not going to talk about that. Correctness is a hard problem. I'm more interested in, was the analysis actually performed as it is described, and can you validate that? And so this leads to this question of what is reproducibility for computational analysis? To me, this means that the analysis has been captured or described in sufficient detail that it can be reproduced ideally by a different person in a different environment, right? So starting from the same data, you should be able to get exactly the same results. And so there are many other concepts like generalizability or correctness that are bigger than reproducibility, but I would say that this is a starting point. This is an absolute minimum standard for what we need to expect from an analysis. And yet, most published analyses are not reproducible. We can quibble about the exact fractions, but there have been a few studies looking at different things. I'll just point out, we did this for 50 resequencing papers specifically in 2012 and found that only about 7 of those could be reproduced. And also there was nice work done with the Cancer Biology Reproducibility Project. So that group has targeted 50 high-profile cancer studies to try and reproduce. And in this publication, another group actually looked at, okay, so not even reproducibility, but identifiability. Can I just determine what software was used? Can I determine what antibody was used? Can I determine what cell line? The answer is almost invariably no. And in particular, for the analysis side, only 32 of 127 tools that were referred to in these papers and the methods of these papers, you could actually figure out what tool they really did use. And that means, you need to know versions, you need to know parameters that are used in analysis. So this is pretty bad. And so that was only about 6 out of 41 papers that were looked at here. And people often say to me, so what? And I just want to tell you that methods do matter. This is looking at different versions of an alignment tool called BWA, pretty commonly used alignment tool for resequencing. And what we have, so we have different versions, we have different parameters, and basically what you're seeing is that your results change depending on the version of the tool you use. The algorithms are changing constantly. They're being improved or not improved or whatever, but results change, and so you need to track all of this information. So the core tasks then I'm interested in are we need to capture the precise description of the experiment. Ideally, we could do this in a kind of transparent way. And we need to assemble all of the data and software and then put those things together to reproduce the analysis, right? So there's this capture task and this assembly task that are both critically important. Okay, so then is this an achievable goal? Well, there's been a lot of work on this. We've done some with the Galaxy analysis environment. IPython notebooks have become really popular and powerful, especially recently. In the R community, lots of work has been done on these literate programming, integrating code into publications. We can think of using virtual machines. And so there are lots of solutions that solve components of the reproducibility problem. My favorite that I'm selling to everybody right now is these containerization technologies that make it super easy to capture all of the software and dependencies used for analysis in a very lightweight way. So this is transforming how we think about packaging and sharing software in my group and with my collaborators, and so I think it's a powerful part of the solution. As I said, these are all solving part of the problem. Well, and even partial reproducibility is better than nothing. I really think that striving for reproducibility inherently leads to transparency and making the research more understandable, and thus any efforts to make it more reproducible are also making the science better in the end. So we put together a few recommendations I'm just going to highlight quickly for doing reproducible computational research. The first is simply accept that computation is an integral part of biomedical research, period. It's not an afterthought. All biology is quantitative biology now. All biology is computational biology now. This is just an essential part. And if you're going to work with data, and it's hard to avoid working with data these days, you need to familiarize yourself with the practices of doing that well. Always provide access to raw primary data. I think at least this is kind of uncontroversial in genomics. We do have questions about how to do that in the safe ways that respect privacy, but at the end of the day, I think we all know that this is what we want. What's missed sometimes is auxiliary data sets. Databases change constantly. This really can affect things like annotation. And so it's really important to ensure that data sets that are used in the analysis are archived. If you have to archive them yourself, go for it. Storage is cheap. It's better to do it than not do it. Similarly, for all software, we actually like to archive the software. You may know a version of a particular piece of software that you use, but what if the place where that software is available goes away, and you want to look back at this paper 3 years later and the software is gone? If you've archived it, and Docker containers would be one great way to do that, then you can definitely recover later. Record all parameters obsessively. This is where a lot of papers fall over, is they assume some parameter is unimportant, it's not recorded, or they assume the default is easily discoverable and then later find that it's not. And this can lead to significant issues in reproducing, similarly for any kind of custom scripting. And then finally, not really a rule for reproducibility, but one that can help us all do better is to use existing software pipelines to try and assemble best practices as a community rather than everything being a one-off analysis. This makes it just easier to understand and inspect and improves pipelines for everybody. All right, so reproducibility is possible. And I'm going to argue that reproducibility is not a technical problem anymore. There's definitely improvements to be made in the infrastructure side of this, but reproducibility really is a social problem at this point. Yeah, it's slightly more difficult to be reproducible than not, but only slightly. We have we know how to do this if you actually want to. People don't know how to do it right. That's the problem. So I think education is actually probably the most important component of how to make science more reproducible at this point. People just need to know that they can do better, and it's not that hard. This fear of being critiqued thing is something I see a lot. These are both things that people, students, postdocs in my lab have said to me. Right? It's like, my code is too ugly. I don't want to put it out there. I don't want to put it on GitHub. People are going to laugh at me. It's like, to me, that just means, okay, you're saying you're not actually confident that your methods are actually correct. So that should not be an acceptable statement. Now, this other one is a bit more nuanced. Why hold myself to a higher standard? I've certainly had people say to me, look, my competitor is not making their methods as transparent and open and reproducible as you. So I'm at a competitive disadvantage now because I've made my science more transparent. I don't know how to fix that one. We just have to do what's right, and we have to not accept people not being transparent. And that's where I think a cultural change is really necessary. As I was saying, we need to create this expectation. There needs to be a stick. We're not just going to incentivize it. We need to expect it. The burden is on the authors, and so peer review is really the gate of this, and people say, well, as a peer reviewer, I don't have time to go through all the work of trying to reproduce the analysis. Well, then say the authors need to make it easier for you to do that. They need to provide easy ways that you can verify that the analysis was done correctly. And education is critical. I'll just point to a little opinion piece from a couple of my colleagues at Hopkins called Reproducible Research Can Still Be Wrong. And basically what you've got here is once your paper is submitted to a journal, there's lots of ways you can gate the process of ensuring that it's quality, ensuring that it's reproducible. But really what we need to be thinking about is teaching people to do data analysis properly. That's how you're going to really fix this problem. So I think, I'll say again, widespread education is absolutely necessary. I think that if we want reproducibility for really complex, data-intensive experiments, they need to be more open, right? Review needs to happen more frequently, continuously, and I'm going to go out on a limb and say that it should happen openly. You're not going to promote open and transparent science in a system that's built on blind peer review, just an opinion. And the most important point I want to make is that mistakes will be made. Mistakes are being made constantly. Genomics especially is full of mistakes. All we can hope to do is find them. So openness, transparency, reproducible pipelines move you to a place where you can actually start to find things that are wrong. And so we need to be a bit accepting. We need to say, look, it's okay if if your analysis has a mistake in it. It's more important that you get it out there in a way that I can inspect it and actually maybe help to improve it. And so that's what I would like to see. All right, just a tiny, tiny bit on correctness. I think I'm under time. Oh, yeah, definitely. So I said I wasn't going to talk about correctness, but I will a little bit. So we have a different kind of question, right? What is the right way to analyze a given data set? And how do we establish best practices for data analysis? This is a figure, it's a couple years old, but I absolutely love showing a kind of a very different Venn diagram than what Jim was showing. So Jim was saying, okay, we use the same methods, but we sequence the sample 3 times. Here, we've got exactly the same sequence. This is a very deep 120x exome data set, and then 5 different analysis pipelines are applied to it. Now, before you saw with a single analysis pipeline, and you could see 99-ish% concordance. Here we see 60-ish% concordance between the variant calling pipelines. Things are getting better. Genome in a Bottle is doing amazing work to provide the gold standards that are going to allow us to optimize these pipelines for different cases. But we really do have a challenge of how we decide which is the right way to call variants. I like bake-offs, open challenges, where we can actually have people contribute methods and evaluate them in a systematic way. And I think that, getting back to reproducibility, having pipelines you can run reproducibly are essential for making fair comparisons. And so one that's going to be starting soon is the ICGC TCGA heterogeneity challenge, basically looking to estimate the number of subclones in a tumor population. And this is all work, and I took this slide from Kyle Elrod at UCSC. The idea here is to actually, that the contestants submit pipelines. But rather than you giving them the data to test on, they submit their analysis pipeline, and the challenge system runs the analysis pipeline on the data and gives them a score, right? This eliminates cheating. I'm not actually that concerned about cheating. It has a much more important consequence is that it ensures at the end you have pipelines, right? You can have a pipeline that you can now use because it's inherent in the evaluation process. And so basically what this ends up doing, it happens to be Galaxy. It wouldn't necessarily need to be Galaxy, but it's a combination of Galaxy and Docker. So you submit a workflow and a description of how to get all the underlying dependencies. And we build that environment inside the Google Compute Engine cloud, run your analysis pipeline and evaluate the results. And so I think this is a fantastic model for how to do this, and I'd love to see more of these kinds of challenges. Okay, that's all I really wanted to say, so we can have questions. Just acknowledge my colleagues at JHU, especially Jeff and Roger, who I took some stuff from, and Anton and Jeremy from Galaxy. Gary and Ivan, we worked on that 10 simple rules for a reproducible computational research paper a while ago, and then Kyle for the ICGC TCGA stuff.
Thank you, James. So there are many sequencing technologies and exome enrichment kits, alignment and genotype calling choices, versions of the reference genome, statistical considerations when detecting variation, technical and experimental biases, and it's all a bit of a moving target as far as how rapidly the field is changing. We might strive for and even achieve reproducibility in experimental analyses and in computational analyses by using best practices, providing access to the raw data, capturing all the experimental analysis details, defaults, parameters, and archiving the software. So I'd like to just talk a bit about how journals promote reproducibility and in particular genome research. So at GR, we find that failures in reproducibility can in large part be mitigated by quality peer review. Reviewer choice is important, And we strive to garner reviewers who are high-level experts, recognized to reliably provide thorough and constructive reviews, and who cover the breadth of topics and technologies reported on in the manuscript. These reviewers know that their responsibilities are to judge and offer suggestions for improving the quality and clarity of the research. They are charged to ask themselves, have the authors used the appropriate technology for the hypotheses tested? Have they included a sufficient number of biological and technical replicates? Have they employed the appropriate statistical tests? Have these results been validated? Are the methods clear and complete, including primer sequences, buffers, kits? Are all software used, cited appropriately, and are the custom scripts provided? While it cannot be expected, the best peer reviewers access the data, sometimes via a reviewer's link or the supplemental material, and spot check the quality or even rerun analysis and or test the software being reported. These considerations should be implemented when you review and be driving your own research and reporting. So we promote the reproducibility of research with a strict data release policy, encouraging all data producers to make their data as freely accessible as possible prior to publication. We encourage all data producers to make their data as, upon publication, reported data must be readily available to the broader research community via public databases when available. When a suitable database does not exist, the data must be available on the Genome Research website and, if desired, on the author's website. This is for all different types of data. Our rationale for this policy is that the underlying data is necessary for the greater scientific community to assess the quality, reproduce the work, and extend the science and make new conclusions. With regard to sequencing data, we do not consider for publication manuscripts where sequence data reported in the paper is not freely available in either a public database or where there are issues of informed consent or identifiability in a controlled access database. The accession numbers are included within the manuscript prior to acceptance and ideally upon submission so that the reviewers have access to these data. We promote reproducibility with the journal standards and editing by ensuring complete methods reporting. In our instructions to authors, the materials and data release policy is front and center, and a statement regarding data deposit is required in the cover letter. It's also clear that a data access section and or software availability section is required. This is prior to submission. We are currently updating the instructions to authors as the editors are in favor of more detailed instructions, perhaps including more specific file formats that are accepted, and considering including a checklist that would remind authors to include such details as the software version and parameters used. During online submission, an author is required to read our data release policy and indicate that their data has been deposited and accessioned, or deposited and accession numbers are pending. With regard to methods reporting, although the reviewers are expected to ensure that it's clear and complete, the editors of GR believe that sufficient space should be allowed such that that can happen, and so that it's detailed enough to allow any qualified researcher to duplicate the results. When the method sections are too brief, authors are told they must elaborate such that non-standard methods should be described with sufficient clarity and detail for the broader readership. The editors also go over these carefully, ensuring enough detail is offered, and there are pointers to the supplemental material where more detail may be found. And any data that cannot be deposited in a public database is included in the supplemental material. We've also not limited the length of our supplemental material because we understand that additional supplemental methods, appendices supporting data that cannot be deposited is common in genomic studies. We expect the source code to be included in the supplemental material as a way of archiving the version that was used in that paper and for the reviewers. And And as mentioned earlier, a data access section is required, and the editors check that all accessions link to the data, that the data we expect is there is actually there, and that is publicly available prior to actually posting online. Where data has been generated or software developed, a software availability section is required with a link to a downloadable executable source code is in the supplement, as I mentioned, and we also encourage authors to deposit their source code in, for example, GitHub, and have a DOI for that. So what do we do when there is a reproducibility issue? First, authors sometimes contact the journal and inform us that they cannot replicate their own work. We publish corrigendum, as David mentioned earlier. These report assembly errors, errors in formulas used or in data analysis programs, errors in the figure presentations. We also contact authors on behalf of a reader who cannot access data, although we do first encourage direct contact with the authors. And further, we have conducted technical reviews to determine whether the reader is asking for data required to replicate the study or for more data than is required to reproduce it. So in closing, the standards for data deposition is a prerequisite for data reproducibility is an ongoing discussion between community members, their funders, and editors. And with that, I'll open the floor to questions.
Can I ask a question of both the speakers together? Jim, you very nicely talked about particularly how when you take the same samples and you put them on different chemistries and different machines, there are some differences. And the question is, what's the relative fraction that differs? Because the hard thing that we face is how we're going to have interoperability when we try and build these larger databases to try and understand things. If indeed the technologies keep changing, we can't be in a position of going back and rerunning everything on the next exome or the next whole genome chemistry. What is your assessment of where we are now with that interchangeability and interoperability from a technical point of view? And then I want to go to James, where you were talking about best practices for analyzing data. The best practices, in my mind, how do they take into account this particular technical issue that is going to be with us forever? You know, we have all of this data that we've generated. We're not going to go back and redo it anew. So, Jim?
I'll try to take a crack at what you were asking. I mean, right, so if you're using different chemistries, even on the same platform, I showed how you can get different levels of concordance. If you change the read length size, compare it to the 2x125 to the 2x250, there's 40,000 differences at a standard threshold that we would use for calling genotypes. But that's in the background of 3.5 million variants in the whole call set. So it's a small fraction, but it's 0.8%, I think it was, something like that was the difference there. So it's significant, and if that's just one sample, and I think what you're maybe getting at was what happens if you do this with lots of samples, right, and compare them, how much does that compound? To some degree, the systematic differences should hopefully also overlap if you do have two separate sequencing platforms. You may have differences between the two platforms. You can kind of break up your two different sequencing methodologies and say, well, these are separate replicates of the experiment because the platform changed, and then you're comparing replicates with one technology versus another technology, even if it's the same platform and just different sequencing modalities. Did I answer your question?
Yeah, I mean, and to go a step further, I know from the GWAS experience where we've used 13 different versions of Illumina chips on literally the same sample, and we've been able to identify a small fraction of what we call our starred SNPs, that represent about 0.5%, that the assays just don't perform well. How much is it, so to speak, the assay as opposed to just the randomness of going when you do this enough times, do you see that should we have a part of the genome that's grayed out that we know that, danger, go there
yes.
with a certain amount of caution? No, but it's important because you don't, how much do we want to fight to get that corrected and at the same time have to do overkill to look at the other 99.2%?
Now that's a very good point. It's interesting, when you look at genotyping chips, they've actually selected those SNPs in regions that are relatively easier to genotype.
Right.
When you do a whole genome, you have all the regions that are difficult, all the regions that may not be captured in the reference, and then you're getting things that just show up because this technology now puts that read on that place, but it shouldn't have been there because the reference wasn't as accurate as it should have been. There's lots more things going on when you do whole genome sequencing. And whole exome is even a little more extreme because you have a lot of what I call edge effects. Every single edge of every exome capture is regions where you're decreasing from good coverage to low coverage, and you'll right on those borders, which you have lots of 200, 400,000 borders, you're going to get a lot of errors building up in those regions. But the genome in a bottle is James mentioned earlier, is tackling getting a very high quality sequence on a few samples that will be distributed for reference standards. And they've actually said, these regions we can call accurately, these regions we can't. You can test your method on the regions that we know we've called really well and see how well you're doing in those regions. James, do you want to?
I think that's the point I would make as well, as gold standards are really the only solution for recalibrating the pipelines as things change. So ideally, we'd have referenced instruments that you could resequence and reanalyze and have a known underlying result. So I'm a little optimistic at the same time. I mean, it's not clear how many experiments you want to do are actually represented by any given gold standard. So that makes me a little nervous. It's a lot of effort to solve one very specific problem, and there's a lot of sequencing and analysis problems we don't have any hope of a gold standard for anytime soon.
Well, there's never enough gold.
Luis Franco from NIAID. I often tell the graduate students and the medical students that looking at a heat map in a publication is like looking at a tree that looks like Mickey Mouse. You don't know if a thing really looks like Mickey Mouse or if somebody trimmed away everything that did not look like Mickey Mouse until the thing looked like Mickey Mouse. And so I think with data that happens a lot, and you mentioned this issue of data cleaning and how that happens. So what we've been doing in the lab with things that we're producing now is to use a fairly simple standard, which is to make sure that we document things in a way that can easily be put as supplementary materials and that correlates exactly with the data that we're actually going to upload to a public repository, which was a problem in the Ioannidis paper that you also mentioned when they tried to reproduce these microarray experiments and they couldn't. And part of the problem was that the things that were actually uploaded to a data repository were not exactly the things that were used, and people had trimmed away things or excluded samples and so forth. So the standard that we're using in the lab right now, and we're using SWeave, which was mentioned in the second talk, is to say we are going to present to a reviewer and eventually a reader of a journal exactly how you go from the data that are uploaded in a public repository to our conclusions. And it's a very simple standard, but I suspect that if a standard like that was implemented, it would be way easier to come up with this notion of true reproducibility. So I wonder what you guys think about such a standard.
I fully support that idea. Right? That's the dream, is that actually I'd like to even go a step farther and say you don't submit your conclusions, you submit the raw data in the pipeline, and it gets run, and that's what the reviewers see. So they're never seeing anything that you've had a chance to cook. I would really like it. And coming up with some open standard for how to provide that would be absolutely fantastic. I also, if you do that, you can do some interesting things that take it a step farther, like live figures, for example. What if you have the pipeline, you can basically tweak things and regenerate figures, making it new ways to explore publications. So yeah, I'm so for that.
To me, it seems like a simple enough standard. How do you go from what's uploaded to what we concluded?
Traditionally, it's been a little tricky because of issues like the environment, right, where it's actually run. But I think those are all solved problems, or almost solved problems now. So I think it's right. The time is ripe to implement some kind of standard like that.
Yes, but I mean, a lot of the times when you're going to go do these analysis, we are adjusting. We are looking at covariates. We are looking at choices of samples and how we're going to run particular tests and whether we're generating heat maps or association tests. And now you're getting into sensitive data and the availability of being able to get the information so that you're going to adjust for smoking or for psychiatric medicines or whatever that are going to be incredibly informative, because genetics is not a 2-by-2 table, as Les very nicely had that right, wrong, and sort of another direction slide. So how do you bring that into this?
So, yeah, well, it sounds like you're folding back in what you were talking about earlier in your talk, and yeah, well, it's you know, if you are, it's much more complicated. You need to get the data into like dbGaP if it's, you know, phenotype information, environmental information as well if it's smoking versus not, or all that needs to be available. If I'm answering what
Right.
you're asking.
Should there be an intermediate point where we would say, to convince the reviewers and the readers that the data is stable, that at a certain point, without all the necessary adjustments and the things that get into the phenotype, that you can at least show that the data structure and, you know, the calling of the genomes and the, you know, the parsing of them against a particular outcome would be done. But when you go deeper into looking at, you know, whether it was a left-handed person who had, you know, a psychiatric disorder and smoke, that gets to be a harder thing to do in the way in which we share data until we can freely have everything available that's already been consented. So, you know, is there an intermediate place, or is that too difficult or not really necessary? I don't know. I'd be particularly curious to hear some of the editors talk about this, just because it gets to sort of data integrity and what you are willing and wanting to get out on as part of a publication or not.
Well, it's related, but not, it's related to what James just said, what I wanted to ask, but not really to what you just said. So if you have something to say, Hillary, on this, go ahead.
No, I'm not sure I understand. So are we still talking about the data behind a figure being generated in particular? Or
maybe I can take a crack at this. Again, I mean, it gets into, again, how much information is just brought to the table by the expert that sits down with the data and all the information, and then comes down to the final conclusion that hopefully we can be independently reproduced by other studies. But there's a lot of knowledge, a lot of background, a lot of information that's really hard to capture in sufficient detail so that anyone can dive into the data in the same exact way.
Okay, I'd like to ask a question of James. I'm at Nature Methods, and I love the idea of, in principle, of the raw data and the pipeline being made available, and then the results being, reproduced, or the question of whether they can be reproduced. But I have to, and it's easy. I mean, it's great to hear that it's a solved problem, but I have to say, from the perspective of assessing new methods, we have a hard enough time getting reviewers to, and I'm not blaming reviewers. I think it's a difficult thing to do. Getting reviewers to use data sets provided by authors to test these methods, that it doesn't seem to me a solved problem at all, at least on the ground. And these are sophisticated. Typically, these are methods being generated by sophisticated people, and also being assessed by people by experts. I mean, if you're talking about people using computational analytical methods and then just providing the raw data and their pipeline, I mean, I don't see how that's going to happen anytime soon.
I guess two answers. The first is, when I say it's a solved problem, I'm talking about the technical problem. The social problem is profoundly unsolved. That's where we're stuck. And everything you say is true. If I get to be completely idealistic and take over, I can come with a completely new peer review process that incentivizes something differently, but I realize it's unrealistic. At the same time, I am completely idealistic, and so I can say that's unacceptable. You can't say I have a methods-intensive paper and it's too hard to provide the methods. It's ludicrous. It's not science.
Absolutely. I mean, providing the method is not in question, but it actually gets quite difficult to have it assessed properly. Still.
Yes, expertise. If that's the concern, having the appropriate expertise to review the methods, I totally agree. we frankly need to spend more time doing peer review, and maybe there's infrastructure we can build to make it easier for people to do this kind of peer review so they can focus their time where it's needed rather than on the unimportant stuff of, you know, getting somebody else's code up and running in your system or whatever. So I agree, but I find the situation unacceptable, and I think we need to invest in making it better. So you're absolutely right.
Just sort of speaking, but I just wanted to make another comment that I believe that this issue of computational and analysis of data being not reproducible is just the tip of the iceberg with respect to the broader issue of non-reproducibility, because the methods themselves, the way that things are done technically in the lab, can also deeply affect the reproducibility of research. And I think that part is not completely, but almost entirely neglected at this point. And I think that, for example, the way that randomization is used in clinical research as opposed to basic research, and in basic labs you still have a graduate student that can choose which antibody to use, and of course the newer antibody usually goes to the thing that should light up brighter. Those kinds of problems are often neglected, I feel. And no matter how much data analysis we do afterwards, if the data were not generated appropriately and then that part cannot be reproduced, then we're still doing science that's not leading us anywhere.
I agree there's lots of challenges at that level as well, but what's been shown, I think, in the large part, is that this methodology, technology is producing wonderful and great, powerful results. But there are all the technical considerations that need to be considered. I hit on some, and I think you're going back even further to, okay, what antibodies were selected and what choices were made. Was that recorded? Are those available?
Randomized?
Those, yeah, right. So I think the last.
Hello.
Yeah.
I'm Jeremy from NCBI.
Yep.
You mentioned about the reproducibility by running the same pipeline. But if I would refer a genomics paper, I would rather much be happy if I run a different pipeline and I can reproduce the results, say, to 98%. It won't be 100%. That's much more valuable. And I think the big problem about why is genomics the least reproducible science around is because when we are given the data, we are supposed to refer a paper, we are usually given 1 week. And we need 2 months to actually reanalyze the data. And we should reanalyze it with a different pipeline. We find the same result, we're happy. The fact that the same program would give the same results, that's not so important. Maybe.
I agree, but I think it's an essential starting point to be able to ask the more complicated question. If the same program doesn't even give you the same result, how are you? What's the point at that point of even evaluating the?
Well, if I can reproduce with another program, then that's good.
Yeah, it is. That would be awesome. I wonder if that's going to happen.
Okay, I think we'll wrap up this session now, and there's a bit of a break. 3 p.m.
The next 2 sessions are actually epigenomics, not just genomics, but they introduce some similar and some different issues as genomics do. And so I will introduce Natalie D'Souza, who will introduce the speakers.
Thank you, Michael and Paul, for inviting me. I'm at Nature Methods, for those of you who don't know. Our session, which has moved from the last session, is the one on RNA-seq and transcriptomics, and we'll have 2 speakers. The first will be Paul Meltzer from the NCI. He is a tumor geneticist who's been working on cancer genomics for a very long time, and he's now the chief of genetics at NCI CCR. He will introduce RNA-seq and talk about the potential and make also some general comments about the pitfalls. And then we'll have Christopher Mason as the second speaker. He's an associate professor at Weill Cornell Medical School, associate professor of computational medicine, although he tells me he's really a geneticist. And he will talk more about problems with reproducibility, or issues with reproducibility in RNA-seq. So after that, I will make a few comments, and then we'll take questions in the way that the other sessions have happened. So, Paul.
Okay, so this has been a really fun workshop so far, and I think we're entering a very exciting aspect of it, which is what I think is a truly revolutionary technology evolution that is part of the sequencing explosion. And I'm going to, in this talk, kind of just introduce what RNA-seq is, what it can do, and the good side of it, and then go through some of the issues that are really related to the topic of this workshop, which is reproducibility. And I think Chris Mason will then go on in some more nitty-gritty details about those problems and his take on that. So, we really are in the middle of a revolution here, and we had a nice settled world of expression profiling at large scale using microarrays. We had already learned most of their weaknesses, flaws, challenges. We had stable technologies. We have stable technologies, reasonably stable software. And then along came sequencing, and very quickly it was discovered that you could, use these short-read sequence tools to sequence RNA. And that's really created a massive sea change with tremendous new opportunities, no doubt about that, and also huge new challenges, which are as yet incompletely solved. And, I think just by way of introduction, at this point in the history of biology, tools for the large-scale measurement of gene expression have become central to biological research. And if DNA, is the book of life, the transcriptome is kind of the movie. So it's really the exciting part where you get to see how the genome functionally plays out, because most of the genome activities, functions are mediated through the RNA world. And we now know that a very large percentage of the genome is transcribed in one situation or another, and we know very little about what a lot of that does. At this point, we also have huge databases of publicly accessible data and pre-computed gene signatures, which are of a value that are far beyond any initial publication of a given data set. So this is one area where the concept of sharing genomic data has really paid off in a very big way. So that's a really important point. And this is just another look at this technology revolution, the microarray to RNA-seq. And in some ways, the microarray world was easier because it was a more constrained problem. It was constrained to just the measurements you could make with microarray probes, even if you had millions of them. The RNA sequencing world is wide open to whatever you can map to any genome. So something exciting is happening when you get this kind of growth in citations, and it's been roughly doubling for RNA-seq every year. Those numbers on the bottom are off a little bit, so it would continue to double. I think it's about 1,700 in 2014, so it's been a little more than a doubling every year, starting from 2008 with these three really seminal papers that reported the ability to get good data, usable data from RNA sequencing technologies. And just for fun, this is Sean Davis did a little bit of a data analysis of what's in the NCBI Gene Expression Omnibus Geo database at the present time. And the two curves I would call your attention to are the top one, which was our microarray submissions, and the bottom one, which is RNA-seq. And you can see this thing taking off, and it's headed up and close to catching up to the microarray submission. So it's really taken off tremendously in the last several years. And so, but it's important still to remember that in terms of using transcriptomic data to address biological problems, there are still 39,000 plus data series in Geo, which are hugely useful, and only about 4,500 RNA-seq sets so far that are available. But this gives me a chance to mention this idea of public availability of the data and to point out that GEO is there with about 1.4 million samples worth of data in it so far. There's another database at EBI, Array Express, which also is storing this kind of information, has 1.7 million individual assays in it. And so you put these two together and you have a huge amount of data. So whenever anybody says, I want to do large-scale transcriptome profiling, I say, are you sure it hasn't been done already? Or at least if it hasn't been, what data do you want to compare it to? See how that was generated and what will be the easiest way for you to compare your results with those results, because that's what they want anyway. So it's really simple. This is like the reduced complexity version of RNA sequencing. You start with RNA, you break it up into little cDNA pieces, and sequence little tags at the end of each fragment with a short read sequencer. And that's the majority of what's been done. I should add, though I'm not going to talk about it in detail, that there are also some long read technologies which are particularly turning out to be valuable for certain aspects of disambiguating alternative transcripts, for example, can be quite useful, but haven't been used nearly as extensively as short read technologies. And of course, the other thing I'll point out already, just to hint at some of the problems to come, is that I've diagrammed this as if there's a very beautiful even distribution of fragments generated in an ideal transformation of a long RNA molecule into a library. But we know that this is not a completely unbiased process in the real world. But when you're a biologist and you see something like this is several different RNA-seq samples plotted and displayed on the Integrated Genome Viewer, IGV, and you can see a gene which, multi-exon gene expressed so definitively, so you can see the expression of every exon. You know for sure that this gene is actually expressed in every one of these samples, and you can say something about the level of expression, the usage of exons, and the presence of mutation. It's just exciting. And so this has really caught fire because of this intrinsic excitement. So, RNA-seq can measure expression levels, but what it really adds are the ability to determine transcript structure, to recognize aberrant transcripts arising from DNA rearrangements, to pick up small sequence variations, look at RNA editing, and even to identify novel transcripts in genes. And I'll just show a few examples of what this looks like for people who've never seen this kind of data before. So here's a melanoma that has a mutation in BRAF, V600E mutation, and there's the RNA-seq, and it shows the mutation at this site that is present in 100% of the reads, and that will tell you right away that there's only one active allele, and that's the one with the mutation. That's interesting if you want to know what that mutation does or decide if it's important in that cell. It's more likely to be important than if it's not expressed as an activated mutation. Here's an example of a transcript structure abnormality, fusion between BCR and ABL, and this split screen view shows the color highlighted reads that show the mis-splicing between these two disparate genes that have been fused onto the same chromosome. So a very powerful tool for detecting these and many more of this cancer type of rearrangement are being discovered through the use of RNA-seq virtually by the month. This is a beautiful example from Milla Prokanina-Olsen's lab where she actually, I think, has had a major impact on the problem of hepatitis C infection by discovering a novel transcript in poly-IC treated cells, which was inducible and turns out to be a new interferon, which was not previously annotated in the genome. And RNA-seq made this discovery possible and also gave it certain initial biological credibility, which I'm sure excited people to work more deeply on this. So that's a beautiful example of a gene discovery. So there's some important issues in transcriptomics. First of all, there's a huge dynamic range, and this is a little bit bothersome. It was very bothersome when we first started out because and sequencing was particularly expensive because we're going, we're sequencing an awful lot of copies of actin to get down to the less abundant genes. That's less of a concern now as the sequencing costs have dropped, but you still are sequencing an awful lot of actin in each one of these experiments. The next issue is that there's significant molecular diversity in the RNA world, RNAs of all different kinds of size and structure, and that and function, and that creates a big challenge in measurements. Not all transcripts are known, and not all genes are known. And then, of course, all technologies are imperfect with platform-specific limitations, which you have to understand. And analytical tool development in this type of situation always lags behind data generation technology development. So we're pretty good now at generating data. We've started to get a handle on some of the problems that are involved, but really analytical tool development is still ongoing. So getting to the molecular diversity topic, mRNAs, of course, was the initial goal everybody thinks of, but now we know much more about the RNA world, and so microRNAs, other small RNAs such as snow RNAs, long non-coding RNAs seem to be increasingly important. There are gene-associated non-coding RNAs which may have regulatory function, and there can be extrinsic RNAs in a given sample that might come from a pathogen, a virus, or something that's not supposed to be there. So that's another thing you can pick up in this kind of experiment. So with this great technology, can one experiment measure all of the above? And the answer is no, no way. Nothing right now can get us all of that information from one sequencing experiment. So the bottom line is that RNA-seq experimental planning requires knowledge of the technology, its performance, and its pitfalls, and the experimental design is absolutely critical before you launch anything that you do. And the protocol selection is simply going to be dependent on the study goals. Now, to get to the issue of QC and all of the issues, I just point your attention to this issue of Nature Biotech from September 2014, which has a number of really excellent articles that address this in detail and take up many of the issues I'm going to just allude to very briefly. So what are some of the limitations? To start off with, there's a lower limit of detection that's constrained by the mRNA or RNA abundance distribution in the number of aligned reads per sample. So in the end, you're constrained by the number of reads you can generate. Large sample numbers at this point are still fairly difficult to process without automation, so it can be somewhat cumbersome to process a large number of samples. The software, as I've alluded to, is still evolving and it requires sophisticated bioinformatics collaboration. The computational hardware and expertise to use it is substantial. The library prep methods are still evolving, and the data comparison problem, if methods differ, forget from RNA-seq to microarray, just in terms of different RNA-seq protocols. Data comparison can be very problematic if the methods differ. And because this is such a rapidly evolving field with so many different variable protocols in play, this is a major issue that's very difficult to address. So looking at the sources of variation, first at the sources in the analysis pipeline itself, there are the samples, the sample type, the sample preparation, the sequencing library protocols, the particular sequencing platform, the computational pipeline. All of these are still evolving and have a lot of diversity in the data that's out there at this point. Focusing first on the wet lab and then on the computational side, the sample type and the preparation, for example, a fresh frozen sample versus a fixed sample, a subcellular fractionated RNA versus total RNA, the specific extraction method, all of those things can be important. The sample quantity is important because you may be restrained if you're only analyzing RNA from a tiny sample in terms of the number of reads you can generate. The sequencing platform, oligo-DT primed versus random primed, ribodepleted libraries, the fragment size that you generate in the process, the strand-specific versus non-strand-specific nature of the library, paired-end sequencing versus non-paired-end sequencing, the specific enzyme and primer cocktails where there are many variations in use, and the target read depth that's selected. All of these are variables which affect the results materially, and that's a lot of variables. So the computational workflow, by contrast, is perfectly simple. You take the raw reads and align them. To what, though? To the genome, to the transcriptome, which transcriptome, RefSeq, EMBL, a custom transcriptome, and then what aligner do you use? Turns out there's more than one out there. But let's just skip over those details for the moment and assume you're doing it. Then you get a bunch of aligned reads and a normalized read count, which is typically reported as FPKM, or fragments per kilobase per million, or some transformation of that kind of number. And that's pretty simple. But, well, is it really that simple? The problem is that RNA is made, typically mature RNA comes from genomically non-contiguous bits of sequence, and that is a problem. It's illustrated in this diagram that you have reads that are in red or at the bottom that cross over two regions of the genome. So that's a challenge, and those won't align well with our genome sequence type aligner, so they require an RNA type aligner. Well, even if you've got something like that, then it's still not that simple. And this illustrates, and these are complexities that I refused to believe in for a few months, and I had to be, beaten over the head with computer keyboards and things to say, no, you can't ignore these things. You really have to deal with them. So if you look at this figure, you can see if you have something that's, let's say, gene A, a simple situation, what's the aligner going, it's pretty simple. If the read falls completely on top of an exon, you'll count that. But what if it sort of extends over the end a little bit? Your aligner might choose to accept it or choose to ignore it, depending on how it's programmed. And in the third example, what if it actually crosses across an intron? And what if it's split across an intron? So the software may handle this differently. And you have, the transcriptome is where, like I said, the genome is exciting when you get to this movie because strange stuff happens all the time. And genes overlap, and you can't necessarily assume a read comes from one or the other. How do you handle that? How do you disambiguate overlapping genes? It's really a complicated problem. And this is just another publication that has looked at some of these issues in RNA alignment from Nature Methods, and shows that you can start by mapping the unique reads, but you still will have to disambiguate some potentially. You'll have to look, what do you do with the reads that fall outside of known exons, and how do you allocate reads which as to coming to it from a particular locus. So it's complicated. And then, the ugly properties of sequence bias, which are remarkably similar in all genome technology. So GC content is a factor that affects virtually all genome technologies and even creates a clear sequence bias, as illustrated from this relatively recent study. As you can see, the GC content perturbing the results. There are also very clever people out there writing software, and there are many different classes of models depending on how they deal with all of these different features and in what order they deal with them. And the results may simply not be the same. Now, once you do get through, remember at this point I've just talked about a single sample. So you've processed the single sample and you get something that looks like this, basically a spreadsheet of numbers. Oh, this is actually several samples. A spreadsheet of numbers hopefully correctly annotated. And now you want to actually do something. It's not that exciting to just have a single, okay, I'll go quick, a single result. So you have to deal with differential gene expression. And there's, again, a whole lot of, all of these complexities accompany that data, and yet now we have another kind of analysis and another layer of analytical methods for differential gene expression, and as illustrated by these studies, several studies that you may be interested in digging into. Here, a very nice study about using R in the bioconductor to carry out these types of analysis. So they're very good comparisons of these different tools, and as illustrated in this Nature Protocols paper, some serious thought has gone into the organization of the data flow. I'm just going to talk about fusion gene detection for a second because it's a good illustration of the challenges. It's kind of like splicing alignment, except looking at two different genes. There's a bunch of tools that people have looked at systematically, and they do different things. I won't go through all of the details, there's not time, but they do different things in trying to get the right answer. What's remarkable is most of them, when given a synthetic data set that truly does not have chimeric genes, generate positive results. And this is an illustration of one of the more challenging problems in correctly detecting fusion genes even today. The last comment I want to make is that most of what's been done in the transcriptomic world until now has been a snapshot of a cell population, but transcription is inherently a dynamic process, so you'd really like to know what's going on in individual cells over time. And the technologies now exist for single cell sequence. They bring to the table their own technical problems, which are substantial, but the potential to do really interesting new biology is there. So my quick take on the future is that convergence towards a limited number of protocols and platforms, which specific project types, is going to happen. And there will be standardized software pipelines for routine applications, such as just mRNA expression. And as more widespread utilization will happen, inevitably, as technologies stabilize, as these things are demanded by journal editors, throughput will improve and the sequencing costs will continue to decline. And so I'll leave it at that point. Okay.
Well, good afternoon. I'm going to pick up right where Dr. Meltzer left off, and thanks for the organizers for inviting me here. So I'm going to go through, speaking of a movie, I have a lot of slides, so this is going to be a little bit like a movie, actually. And all my slides, though, are on Twitter. I tweeted all the slides, so they're all publicly available, and just at Mason Lab. So I'm going to do a quick talk about a lot of those, that compendium of Nature Biotech papers. I'll present a lot of those results today. And I'm going to try and do this basically in 4 parts. First is a little bit of background, and then go into some of the data that we found. So the first comment is that people still have this concept that the exome is like 2% of the genome, or you make charts that look like this, and people say, well, we're looking at just the exons, or the exome that people sequence is usually around 60 to 70 megabases, which actually, is in light of more recent evidence, we know that a lot more of the genome is probably transcribed. If you look at the ENCODE paper, it's maybe as much as 80% of the genome has some function or might potentially be transcribed. And so really has, really made that number much larger. Of course, some people have disagreed with this number. Notably, Dan Grauer has this very angry sort of paper where he thinks that it's much lower. He thinks maybe 10%, but I think we can all agree it's probably, he thinks that textbooks dealing with marketing, mass media hype, and public relations have to be rewritten, and that those are the textbooks that have to be rewritten, not genetics textbooks. So probably, if you haven't read this paper, it's in some ways kind of the most hilarious paper to read in terms of just language in a paper, critique of a paper. But no one disagrees that the number is probably bigger than 2%. It's probably at least 10% that in some case active, maybe as much as 80%. And to point out, actually, most recently, the current gene annotation from GenCode, which is probably one of the more conservative gene sets, is really 20,000 coding genes, actually about 25,000 non-coding genes that are active, and many other pseudogenes and also some immunological-related genes. So there's really 60,000 genes. So I often, when I give an RNA-seq talk, I will threaten the audience. I will do it again today. If I hear anyone say, like, oh, there's, of the 25,000 genes in the human genome, I may tackle you or do something or throw, paper airplanes at you. I might do something at you. Because these are these have been well validated and annotated by many, many groups already. So it's at least 60,000. And if you go back in time, actually, Quackenbush thought maybe it was 120,000 genes back in the year 2000. And I did a quick chart a couple of days ago of just what's the number of genes that are actually in the GenCode database per year, per release. It's going up at about 2,600 genes per year. So by the year 2030, we should get back to over 100,000 genes. And we may have no protein coding genes left by then as well, based on these current statistics. So anyway, there's a lot of genes. It's very exciting. Everyone's very excited. We can look at all these genes all at once for whatever number you want to pick. And so you can actually, as was just described quite well, you can actually take a single sequencing data set, look at genetic variation. You can look at differential expression by gene, exon, by splice isoform, by allele, by transcript. You can look at non-coding RNAs and new genes, gene fusions. You can also look at poly-A site changes, microRNA binding sites. And if there's a virus or some other presence, you might actually see it. So it's very exciting. Everyone wants to do all of these things with every experiment. It's amazing. But of course, there is some noise. There's some problems. So in particular, what's really haunted me since 2007 is this problem of the wiggles. What are the wiggles? Where are they coming from? What makes them, essentially? So there's a long list of reasons. I could spend a whole day just talking about all the possible technical sources, actually, which has already been covered quite well, so I'll jump through these. But of course, there's many types of RNA as well. There's at least 26 kinds of RNA that you could be looking at one time. And there's other questions. So, which sequencer should you use? Optical sequencing methods or electrical sequencing methods? There are many ways to sequence things. So that's another question. And also, if you really want to look at all of the transcripts, can you even do it? So if you think of a simple statistic, if you have one exon and one variant, assuming it only gets spliced one way, you could have one thing. But if you have two exons, you could have three variants. You can have then 3 different products. If you have 3 exons, you can make 7 variants. And likewise, 4, you can make 15 different isoform combinations. And at this rate, you can actually make 2 to the n minus 1 in terms of the number of possible variants you could make for any given gene. That means actually if you add up all of the genes in the human genome, there's more potential transcript combinations than there are atoms in the universe. And so if you were feeling at all mundane when you woke up this morning, or maybe you're in the shower and you're like, I'm just a human today, you're actually a very complicated organism, and you can have an extraordinary amount of complexity. But, of course, you can't make all of those transcripts because you don't have enough atoms in the universe to do so. So it's exciting to make all these potential transcripts, but how do you study them? And so the best way, is to sequence them, actually. So, okay, so people want to sequence. We're excited about this. We're no longer sad in the shower. What can we do? So there was a call, actually, by Nature Biotech to say, it would be great if we had something like the MAQC, which had this problem when there were proliferation of array technologies, to actually say, well, we need some way to have a real gold standard, some way to test them. What's reproducible, what's not, how do we really examine this? And so this kind of helped us launch with the FDA, a consortium with Lei Meng-Shi and John, Danielle, Terry Meg, and some other people in this room, this consortium to try and look at this question. So it did at the early stages involve a lot of pilot data, including some helicose, and it's actually a little bit sad because helicose is kind of like the Betamax of sequencers. Like, it was actually really good data, and it just never made it for really market reasons. But it really, as we all know, VHS was not really better. Betamax was better. But in any case, there were a lot of studies, a lot of platforms. We wanted to do the same kind of idea. Another group that I worked with was the ABRF to actually do the same kind of study with other platforms as well. So there's another study that's ongoing now is looking at DNA sequencing reproducibility, also with the Genomes in a Bottle Consortium. There is some work on extreme microbiomes and including some stuff in space that I won't talk about today, but it's very interesting. And so the goal is to do for all these methods cross-platform, cross-protocol, and cross-site, and get a sense of that reproducibility or lack thereof, and to really improve the detection of these signatures so we can actually do this reliably across sites. So the samples that we used across all of these studies, which are in that issue from last September, is A, which is a mixture of 10 different cell lines, and B, 23 different human brains, male and female, varying ages, and A and B. So comparing A and B, they couldn't be more different. There's cancer cell lines and mixed brains. They're very, very different. So it should be an easy case to do differential gene expression. When we published all these data, I was very happy from the previous talk. James was talking about is that, we put all the methods out there, the raw data. We also put all the different detailed methods. A lot of these are also in the paper, but also the scripts. Every single shell script that was run on the cluster that ran every job. It's not quite as good as, say, Galaxy reproducibility, but at least we put everything up there, which was something I was very happy about and sort of proud as a lab that we got out there. And also, the methods that we used to process the data were done by a tool called RMake, and so I'll present some of these data today. So what did we see? So, okay, that's great. It's wonderful. Now, what's the take home? So from this issue, what we looked at is every kind of thing that could go wrong or might go wrong or does go wrong with an RNA sequencing experiment. So there's a whole special website if you want to browse it. It's a very nice website. That's very exciting. You're meeting me. This is everything else in my life will be downhill. I'll never have this many issues or many papers in Nature Biotech again. This is it. You'll see me, crying 2 years from now. This is it. You're meeting me at the peak. The other thing is to note is that we talked a lot about quality checking, and this has come up very recently on Twitter and some discussion about Faculty of 1000 Research where you have Gillet's talking about data reproducibility, RNA processing, informatics processing. So, this has created a bit of a fissure in the field about what people believe. And so regardless of your opinion, it's worth reading the discussion of how people are thinking in a very public form, the processing of data, everything from the analysis to the extraction of the RNA. It's a good read in terms of what the field is actually kind of checking itself for making sure that data is reproducible as a sidebar. But we also looked at these data to try and understand this for sources of variation, how we can solve them. But first, let me get to the data itself. So to go back to the experimental design, it's samples A and B. We spiked in synthetic RNAs from ERCCs, and they did samples A and B, those brain for B and A for the cancer cell lines, and then a 1:3 and 3:1 ratio of those samples, sample C and D, and then again pure spike-ins. And so took all them, and they'd been sequenced between all the different consortia on these platforms as well as solids. So we looked a lot at questions of inter-site variation, as you can see here, as well as inter-protocol variation, and also looking at degraded RNAs and also looking at length. So we had to make this table because one of the reviewers requested, what is the cost per platform? This was infuriating to make because as soon as you make a table of cost per base pair, it's immediately out of date. But nonetheless, the point is, and you notice we don't have any weaknesses for any of the platforms. Everyone has strengths. It's a very happy study. That everything has its different application. Long reads are good for some things. Short reads are better for quantification. I'll talk about this shortly. But we've updated this table, and this is in the slides of, if you take all the different platforms, the cost per base pair, of course, has changed since last September. And if you want to debate some of these finer points, you can. But I just put it up here to update these numbers. So the first thing we looked at was inter-platform performance. How do they compare between different platforms? So one thing we looked at was these different samples up here on top. Was the RNA intact or not? What was the protocol used for looking at them? A poly A prep or rhabdopleated, which I'll get into shortly. And then the coverage, 5' to 3'. If you take all of the genes and put all of them into 100 bins, regardless of their length, and ask the question of how well are they covered, 5' to 3', you can see this is sort of the percentage. You want it kind of to be an even color across the different platforms. And you can see that most of them do a pretty decent job. In particular, PacBio did very well, but that's a little bit because it has a 5' guanosine cap antibody plus poly A priming. So it was enriched to actually get full-length transcripts, and it was sequencing long reads. So this is not too surprising given what the protocol looked like. But the other things we want to look at is between really sort of high-depth platforms, between the different sequencing sites, what's the average coefficient of variation? So this gives you a sense of, in any given platform, they're all expressing anywhere from as low as a 10 or 12% coefficient of variation to as high as sometimes 30, 35, 40, or in some outliers will be higher. And so there is an unavoidable state of inter-site variation within the platform. And then between platforms, we also calculated the R-squared as sort of their correlation coefficients. And you can see high-depth platforms have, a lot of cases, the highest correlation. So they're counting the same genes, although we did use the same genes for all these plots. And so it just means you're getting more looks at the transcriptome. So like Illumina and proton data would correlate more with each other. You're just getting better quantification as you've sequenced deeper. And so other thing we want to see is, okay, well, if you sequence more, do you see more things? And one of the other figures from the SCQC paper is, yes, if you sequence even 12 billion reads, you will always find more things. The question isn't, did you sequence that? It came from somewhere. But is it real? Is it relevant? Is it biologically meaningful? Is a different question. But I can tell you that on all platforms, the more you sequence, the more you will find on all platforms. And also, the splice junctions detected, though, will not be the same let me do this actually, will not be the same. If you have shorter reads here, you can sequence a lot more, but your splice junction detection will go down because you just have shorter reads. I'll get back to this here. So then we wanted to calculate what's the sort of number of junctions detected per million reads, sort of what's the efficiency per platform. And we could see here that PacBio, even though it's not the cheapest way to sequence things, that's still true today. Oh, well, 454 is worse. It's, you do get, as a function of the per base sequence, you get the most junctions detected. And the concordance between platforms for known junctions was much higher than those that are novel, but still we could actually detect a lot of potentially novel splice junctions. So what's important about the splice junctions that are novel especially is if you really want to find novel things, not surprisingly, you need to sequence deeply. This is because you're counting essentially marbles out of a bin, and you have to sample enough of the marbles to find that rare marble that you're looking for. So for the high depth platforms here with Illumina and Proton, you will pull out these rare isoforms. These are called sashimi plots. And so if you're hungry for sushi by the end of my talk, that's not unnatural. The other thing we wanted to look at was the differentially expressed genes. The main reason people still do RNA-seq is I just want to look for something that's different between these two samples. And so we looked at the concordance here, so this another funky Venn diagram of these two protocols on Illumina, and then also looking at the different other platforms here. You can see that in general, there's general agreement, 88 to 97% agreement between platforms for differential expression. For absolute quantification, it's a little bit more of a disaster. But for differential expression, it's usually pretty salvageable, as you can see here, with some degree of disagreement. But this is sort of the Venn diagram you can see here. And so these numbers kind of summarize it per platform. I wanted to put in the paper a proportional Venn diagram, which would have looked like this. This is, it adds, I think it's pretty, but it doesn't actually help you understand the data anymore, but I still like this one better. So the other thing I wanted to look at, since they're mostly comparable for differential expression, but not as much for absolute, what about on the same platform different protocols? And so there we looked at a poly-A prep and a ribodepleted protocol. So one of the first things we observed is when you do a ribodepletion, you're taking all RNAs that either have just been transcribed, maybe nascent RNAs, in the middle of being degraded RNAs, and you'll see all of them. So this is why we see a large pulse of intronic mapped reads, that you're seeing all RNAs that maybe haven't even made it out of the nucleus yet, and you'll see a lot more reads mapping to introns here, whereas for poly-A prep, you're enriching for sort of mature, sort of more grandfatherly RNAs that have been around and know what they're up to. This is these different fractions you can see here. So this has some implications on how deeply you need to sequence. So for example, if you want to look for variants in exons, ribodepletion, you'll have to sequence probably twice as deep or 3 times as deep to do RNA-seq variant detection. So that's a consideration for design. But we wanted to ask the question of the FPKM differences between poly-A and ribodepleted. You can see the log-2 ratio is mostly near 0, so they're mostly the same. But of course, what most of us all love is tails of distributions. There are some differences between them. So what are those differences? This is one of the supplemental tables. Actually, a lot of it is link RNAs and SNO RNAs and small nucleolar RNAs that are actually the biggest differences between the two preps. So if your goal is to hunt for link RNAs or SNO RNAs, a ribodepleted prep is a good way to do it, at least as demonstrated from these samples. But other categories of genes stay mostly the same. And you can see this the table of what exactly those genes are here in the paper. So the other thing is, well, how did they compare? If you do differential expression, do they have the same differentially expressed genes here in green, or are they different between the preps at different fold change and different FDRs? You can see they're mostly the same in terms of the shared DEGs are the same that are detected between platforms, but there are differences. In blue and red, you can see there are some proportion of DEGs, or differentially expressed genes, that are unequivocally prep dependent, that you only see on one and not the other. And then we compared this to TACMAN data, essentially doing an MCC of what is the degree of accuracy. And so in most cases, poly-A was a little bit better, but in some cases, ribodepleted preps were a little bit better for comparing to TACMAN data. And so in this case, you can see, it depends on your threshold, but in general, they're mostly comparable, but there are some differences that are present as a function of prep. So what about degraded RNA? This is the thing that we all, if you've ever worked with RNA at the bench, this is the first thing you fear. You're taught that there's RNases on your skin, on every surface. You're afraid of RNases everywhere. You look at people suspiciously if they're anywhere near your bench because you're afraid of degraded RNA. But one question we wanted to look at is, well, there is a ribodepleted prep, which actually if you think about the prep for RNA-seq as it is, you actually take beautiful intact RNA, and the first thing you do is add divalent cations, and you fragment the SNOT, all of your beautiful RNA, and then you do cDNA synthesis. So it gets degraded anyway as part of the prep. It's just normally it's intact to begin with. But actually, if you look here, these are the degraded RNAs from the samples, both with heat, sonication, and RNases. We purposely degraded them and then sequenced them. And the coverage 5' to 3', you can reconstruct it if you ribodeplete instead of poly enrichment. So we actually see that the degraded RNA actually looks great. You can see here in different samples, different sequencers, different sites, we got the same result. At one point I had a line in the paper that said degraded RNA looks as good or better than intact RNA, and people thought we got upset by that. So now it just says as good as potentially. But you can see by coverage plots it's true. So if you do the R squared of what's the correlation coefficient of intact RNA versus degraded, you can see here in most cases they're very well intact if you ribodeplete it. So there's one way to salvage degraded RNA if you have that problem. So another question we wanted to look at in the sort of last few minutes I have is how do you, so say you have, there is some variation. Two questions really is how can you detect it and how can you maybe get rid of it was the focus of a different paper in that same issue. Was to say, well, okay, every site had 4 replicates. These are the SCQC samples sent out through the FDA study. And there's also, we included a 5th replicate. So every site, they made their own 4 libraries, but we put 1/5 library at 3 sites and said, okay, this is a vendor-supplied library. So the library is the same, and then we're going to send it out to be sequenced. And other sites, they make their own library from the same stock of RNA. So how reproducible was it? And actually what we could see was basically, really, I think, exemplified by this problem, is that if you have A versus B, whether it's a microarray versus RNA sequencing, if you're comparing A versus B, remember we did all the same samples at the same sites, you should see lots of differences between cancer samples of pooled tissue and brain. You do see lots of differences, both platforms. But this concept we had is like, essentially, what about C and D? We should see less differences. So, because remember, C and D are mixtures of A and B, so that it should be attenuated, the number of differences between sample C and D, and indeed that's what you see on both platforms. But the really simple thought experiment we had was, well, actually, what if you just take A versus A? It's the same sample. We actually know that the number of differences between these two samples is 0. It's the same sample sent to the same sites. And so you can see what's interesting is that this gives us an empirical measure of the FTR. And we could see that there were still, between all sites, different sort of areas where you get a lot of false positives. Because we know they're false positives. There should be no differentially expressed genes. These are the same samples. And so this gives us a way to measure what is the actually degree of sort of an empirical measure of the false positives. And so what we started doing was looking at different pipelines, some like the MAGIC pipeline or RMake or other platforms and other pipelines. And it really wasn't sort of an issue of what processing you're using. It's just inherent to the fact that there is, you need to some degree to put a threshold on both the degree of change as well as the minimum coverage that you're seeing. So here's an EFDR across different pipelines. You can see they all have some degree of what's the % of false positives. And so this wasn't just also for sample A. We looked at A versus A or B versus B or C versus C. Take the same sample at different sites and just compare them. Again, we would see anywhere on, this is a log scale, anywhere from dozens to thousands of what are called differentially expressed genes from these pipelines. So then we thought, well, okay, there's other tools out there to try and fix this. So EDA-seq or CQN do GC content correction. Other ones worked on, use this in ERCCs, the spike-ins, to try and correct this. This is a wonderful tool called Remove Unwanted Variation. I love that name. And also other ones use latent variables to try and detect what's present in the samples and try and remove them. So we actually saw here, you can see the original data from Limavum and different tools. In some cases, it helped. In some cases, it made it worse, actually. So if you were trying to actually get rid of false positives, it actually didn't always help you in some cases to use these tools, what we showed. But the real question we want to get at, and so you can threshold this to some degree to get rid of it, but the question is why? What's driving that variation was what really the reviewers wanted to know when we kept going back and forth. So we looked deeper into it and say, we could see that there were certain sites that would get more false positive. In this case, this one site was really essentially getting a lot more false positives. But you can see here, this 5th replicate is down here in green, was not affected. That was the library that was sent independently to each site. And so that indicates here that the prep was the biggest effect here because the library here did not get affected when you look at that one on all these different samples. And so what if we look at the error rate of that sample? Here, the library that was even shipped to independent sites, the square, was still affected because it's a sequencer effect, because the sequencer had a higher error rate at this site. In other cases, though, the gene body coverage, again here, the library is not the issue. So it's a prep effect. You can see it here, but there is some different variations of that gene body coverage. And the nucleotide composition, you can't see the dotted line that well, but again, this is a prep effect, and that is because it's what's driving sort of this noise here. So in the end, we actually validated 2 things that have been known and added 2 more of what is sort of the things that can drive variation. So it really is, you can measure GC content, gene body coverage, the error rate, and nucleotide composition, and tell that in most cases it's a library prep effect. So this isn't surprising to anyone who's done large sets of data before. You probably shouldn't have many sites prep a lot of samples, but just in case you needed to know, is a good reminder. Okay, I'm almost done for time. So actually I'm going to close on just a couple of forward-thinking thoughts. There's a lot of people working on these best practices. There's many people, and there's a lot of stuff online. I just presented some of it, so there's many groups to look at. And then to really close last minute on the future, I think I'm just kind of excited that there is, if you like conference calls, there is a place for you in this world. You can join the Genomes in a Bottle Consortium. The ERCC's version 2.0 is being started for RNA standards. There are lots of standards groups actually working really hard, really collaboratively, really openly. So I think it's never been a better time to have tools and physical standards and informatics standards to really tease out these questions. And so it is here that we can start to address it. And then finally, I'll just do this really fast because I'm out of time, but we should not think just about RNA, but also the base modifications. RNA has at least 113 known base modifications. You can see DNA modifications with PacBio, and we've showed that you can actually see RNA-based modifications. You can take an RNA template and a DNA primer and watch cDNA synthesis in real time and see base modifications. We can see it now potentially on nanopores, so I'm excited about these in the lab. And so if you've not seen this also in theory can do direct RNA sequencing as you watch DNA or RNA move through a pore. The data looks like this. It's a little bit noisy right now, but it's getting better, and we've seen over 100 kb reads, and in some cases, really quickly, you can get two-dimensional reads that are on average of 5 or 10 kb without trying that hard. So we're kind of like this, and we're piloting out direct RNA sequencing. And so soon, the scene in this movie where you thought, Uma Thurman here kissed Ethan Hawke, and she said, well, I don't know if I should date him anymore because I want to look at his genetic code before I decide whether to date him. So she went to a booth and had her lips swabbed and got his genome sequence within 5 minutes. And people say, well, that's crazy. This was in the 90s. You're never going to sequence a genome in 5 minutes. But in theory, if you had a room with nanopores, you could actually do an entire genome in about 2.6 minutes, assuming that they move at actually 10 kb per second, which is not impossible, just a little hard. So at the end, I want to thank everyone in the lab, and thank you for your time. Happy to take any questions.
We are actually running a little late, so I don't think I will say too much. I think I want to leave time for questions. I will just mention there were some very good comments made by my colleagues at other journals in terms of the role of journals and publications in helping solve or ameliorate this problem of reproducibility. And I want to mention just briefly something that some of you may have come across either as reviewers or as authors, the reproducibility initiative that NPG, All of Nature, has been has put into place a year or 2 years ago now. And that's this a checklist that we ask authors to fill out and reviewers to comment on. It's a reporting checklist more than anything else, but it's meant to increase the attention that is paid to a whole lot of aspects of experimental design and execution. I think the best thing to do, since we only have about 10 minutes, is to go on to questions. Does anyone have questions for the speakers on RNA-seq and transcriptomics? If you will come up. Well, maybe I can start. You both mentioned that the technologies, either on the wet side or the analytical side, aren't really coalescing. There are a lot of different possibilities, and the biological question should really drive the study design and what protocols are used. There are so many different variables. If I'm somebody going, wanting to do an RNA-seq experiment to study my sample or to study the effects of a certain treatment on gene expression in my sample, I'm not sure this question can be answered, but what are the main parameters you would recommend they think about as a starting point?
Well, I have to say the most important thing is to really understand what the goal of the project is. And if it's a differential gene expression, then actually the most important point is in the selection of the samples, the number of samples, the number of repeats that are going to be done. And in terms of platform, people, I think, are often going to be in a little bit of a bind because they'll probably end up using what's locally available to them, and they'll be somewhat constrained. So it's not very easy to impose a model. But I think to the extent that they can stick with commercial library preparation kits that are as complete front to back as possible, that will increase the likelihood that they will generate data that will be comparable to other data. That's about all I would say at this point. but there are a lot of nuance to that decision, which is very project specific.
I guess I'd just add that definitely more biological replicates is always better. That's true in science in general. But then, longer reads are better for splicing. So if you're not interested in splicing, you can just do 1 by 50, and that's enough. But then also the annotation is something that I didn't talk about. I talked about briefly, but it couldn't be more important. Actually, my favorite annotation is still the ACE-few gene annotation, which is curated by Jean-Daniel Thierry-Mig right there. And so in the SCQC paper, we said, it's really important. You'll miss, there's at least 60,000 genes, probably more likely, 70, 80,000, depending on your annotation. And you could miss what could be a very important differential spot in your genome because you're just using RefSeq or a lower, a simpler annotation. So annotation is very important when you get the analysis.
That's a very good point. Are there other questions?
I have a question about single-cell RNA-seq. So it was mentioned, but not really discussed. And obviously, there are many errors associated with that. But one of the main issues has to do with the fact that we're being told that, for example, in a tumor, every cell looks very different, and that by crushing the tumor and analyzing the whole RNA, you're not really getting a picture of what's going on. So I have several questions about that. One is, first of all, is that really true, or is that a problem with replicates from cell to cell? And two, how do normal tissues look? Do they show the same kind of variability from cell to cell?
Yeah, so I didn't present any of this, but because we by email, we thought, it's a very nascent field, but very exciting. And so normal tissues do actually have that same heterogeneity, but you will see there is for sure a technical issue with the library prep. It's a lot of amplification. It's using, in most cases, the CloneTech kit or on a Fluidigm or even DropSeq. But you will see, when we've done a lot of single-cell work, they still cluster by patient and by if it's before or after therapy, for example. The biology, it's almost, even though the cells are like a swarm of gene expression, which is very noisy, the clouds are independent depending on who you are or what stage of the tumor you're in. So I think there's actually a lot of hope that they still separate very clearly, at least by PCA, that they're very different. And so I think it's very new, it is noisy data, but they're very separatable actually between patients.
Yeah, and I would just say, I think of it a little bit like flow sorting, so that you would not just compare one cell to another, but a bunch of cells, and hopefully you get some sense of reproducibility in categories that might emerge.
I would just like to make a remark. Chris Mason rightly showed that the library preparation is important. Now, when we are given, say, 400 samples in a big experiment, we can usually recognize the day of the week the library was prepared. We can recognize the technician who did it. And so what you should absolutely do is randomize between the samples and the controls, so that as many samples and controls were prepared the same day. Otherwise, that's the main difference between the samples and controls.
Yeah, that's a hugely important point that gets overlooked repeatedly in this kind of study. It's randomization of the analysis of the specimens after the experiment is designed is critical to get rid of that kind of bias.
Are there other questions?
Yeah, you just mentioned the reanalysis of the data, and I have a program that runs to analyze the whole database for microarray. For me, because there's some biological meaning for each experiment. So how can you analyze different studies, compare their common features or common findings? So how can you, if you compare different studies in the database, how can you set up the common standard to initial analysis, like normalization? Different experiments may use different kind of ways to normalize. Is there any way that you can suggest when you analyze the whole database, how can you make the data set comparable?
Some of it was in the paper. I mean, I think it's always hard because people may have done it in slightly different ways or different protocols. But one of the tools that performed very well was PER, P-E-R, that did well, at least by our comparison, to combine data sets or that even had covariates embedded in them. But it's a hard problem. At some point, you may have to look at data and say, either if the mapping rate is too low or the error rate's too high, you can say maybe this data is not good to use for this big study. But there is so much data in Geo that should be used and could be used. But you will have to look at mapping the base quality, and you can use some of those tools like we published to try and get through it.
Okay, I think we have to stop and go on to the next session, so thank you very much.
Okay, so we're up to our last session. I introduce myself. I'm Kyle Logan. I'm an editor with Nature Genetics. Oh, that's fine. And so the last session is on ChIP-seq and other epigenomic tools. So in contrast to DNA sequencing, where we're analyzing the DNA sequence itself, RNA-seq, where we're analyzing the RNA species being produced from the DNA, ChIP-seq is analyzing the proteins that are bound to DNA, analyzing where they're distributed, where they're bound across the genome, or in the cases where proteins that are bound fairly stably to DNA, what types of post-translational modifications they carry, and how those are distributed across the genome. So it's in some ways more dynamic than the DNA sequence. It varies as a function of the cellular state, varies over time, and our ability to analyze it and assay it is also constrained to some extent by the reagents that we have available to us. In the case of ChIP-seq, availability of antibodies that will specifically recognize the protein of interest. And then there are, of course, a lot of sources of variation in ChIP-seq data and other epigenomic data, and certainly availability of tissue of interest and tissue that's relevant to the question that you're trying to ask, to large part, determines what types of questions you can ask and what types of answers might come out of it. So with that, by way of introduction, we have two very distinguished speakers to talk to us today about ChIP-seq and other epigenome methods. We have Keiji Zhao, who has pioneered a lot of work on ChIP-seq. He's going to talk to us primarily looking at the experimental aspects of data generation for ChIP-seq. And then Shirley Liu from Harvard, who's going to focus more on the analytical aspects of analyzing ChIP-seq and other epigenomic data sets. So turn it over to Keiji.
Thank you, Kaya, for the introduction. As mentioned, that this session will be focused on the ChIP-seq analysis and the related epigenomics tools. And so first of all, why epigenomics? So basically for the mammalian genome, so we know that the DNA, double-strand DNA is wrapped around histone octamer to form the nucleosome core particles. And then this forms the so-called base on the string structure. So this is a kind of a decondensed, accessible form of the chromatin. So transcription factors and other regulatory enzymes that use DNA as a substrate can access this kind of chromatin format in the cells. But by incorporation of linker histone H1, this form of chromatin is condensed to this kind of 30-nanometer fiber that is not accessible. So this kind of organization is really essential for gene expression in the cells. And the basic fundamental unit is the nucleosome core particles, which is crystallized, and that shows the interaction between the wrapping DNA and the histone octamer. So this structure is heavily modified, and as shown here, then particularly the histone tails, so it can be modified by all kind of different groups like acetylation, methylation, phosphorylation, or ubiquitination, depending on the nature of the modification and also the location, position of this modification. So this can have a different consequence to transcriptional regulation. So in cells, gene expression actually is controlled by this kind of various different modifications and transcription factor binding. We know that in mammalian cells, there are more than 3,000 factors, transcription factors, and the chromatin modification enzymes. So the question then, to understand how they regulate gene expression. So as a first step, we really need to know when and where are the histone modified, and also when and where are the thousands of transcription factors bound in the genome. So the best technique so far still the chromatin immunoprecipitation assays to address these questions. So the initial, the very first step technique to do this is a combination of a chromatin immunoprecipitation with the PCR to look at a few genes to see whether a transcription factor or his modification is localized to a specific genetic locus. And then a chip combined with a serial analysis of gene expression, and then can address basically the whole genome. And this is based on Sanger sequencing. As you can imagine, that is tedious and not really efficient. And then when it comes to the next generation sequencing, so the combination of a chip with the next generation sequencing really transformed the field. Basically, in 2007, then 4 papers just reported this technique. And then following this technique, then there are numerous other techniques that based on the next generation sequencing that has been developed to analyze the various aspects of the epigenomes. So this is basically one representative view how the modifications or transcription factors bind in the genome. And for example, this is a human CD4 T cells, and the active regions containing like standard 1 and standard 4 gene. And then this is an inactive region. As you can see immediately that these different modifications can be localized to different regions in the genome. And also this is a silent region, this is an active region, and polymers 2, and CTCF. So I'm going to then describe some basic considerations and then about how to obtain meaningful and reproducible and also high quality data related with the ChIP-seq. So basically, this is the basic view of the ChIP-seq protocol. The first step is the chromatin preparation, and then after you prepare your chromatin input, you do ChIP, and then following ChIP, then you prepare libraries, and then finally sequencing. So this can be used for both chromatin modifications or transcription factor binding. So I'm going just to go through step by step of the protocol to point out the potential issues during the process. So the first step is the chromatin preparation. So the very first step is basically cross-linking. And then the chromatin immunoprecipitation, library preparation, sequencing, and final data analysis based on this. So for sample preparation, and this is the very first step is cross-linking. And depending on your purpose is to analyze the histone modification or transcription factor binding, so you may need to cross-link, or you can also use a native chromatin. So for histone modifications, you can use either cross-linked or without cross-linking the chromatin. But for chromatin binding proteins, you always need cross-linking to stabilize the interaction between transcription factors or chromatin binding proteins and chromatin. So the typical conditions used is basically about 1% of formaldehyde for 5 to 10 minutes. However, so this seems very simple. However, there's a recent paper that tested the conclusion from a recent paper saying that basically in the cells, and then any transcription factor can bind to any open chromatin. But that seems that suggesting that ChIP-seq is a really non-specific tool to see just non-specific effects. But if we you look carefully the conditions used for the papers. So basically, the crossing the cells with a 1% formaldehyde for 60 minutes. So basically that generated really artifact. Anything can be crossed non-specifically by this condition. So specific conditions is really important for accurate conclusion. And so this is the cross-linking. And then after this, you need to fragment chromatin. So that the choice of fragmentation methods also depends on what purpose of the experiments. For his modifications, you can use sonication or like a micrococcus nucleus digestion. And for chromatin binding proteins, so sonication is the best choice. And because micrococcus nucleus digestion favors preferentially degrade linker DNA, therefore, and transcription factors can be bound to linker DNA. So this method is not good to looking at the transcription factor binding site. And then after you fragment chromatin, then you also need to think about what's the size range you need to fragment your chromatin. And so this varies a little bit. For his modifications, usually like a mononucleosome size DNAs is always good, and from 100 to 300 base pairs. For transcription factor chip-seq, then the similar size should also work. But for some large chromatin binding complexes, like a switch-sniff complex or something, and usually you want to have a slightly larger fragments, because if you sonic too much, that may also degrade or break your protein complex. So you may lose epitopes. So that's why a slightly larger fragments is really preferred for this kind of complex. And so this kind of sonication, the size is important, because if you look at the well-fragmented chromatin, like you just sequence the input, then usually what you should see is like a very evenly distributed sequence arrays across the genome. Like this is input 1, input 2, and then this is a H2Z chip-seq. You see very sharp peaks across the genome. But if the sonication, the fragmentation is insufficient, then that can lead to kind of artifacts. And because fragmentation usually occurs at the regulatory regions, and then that generates small fragments that can be sequenced preferentially by this kind of next generation sequencing platforms. One example, for example, in this paper, this is the input is kind of input is using IgG. As you can see that even the normal IgG, non-specific IgGs, pull down the chromatin kind of peaks. And so this is not, so the problem is that the fragmentation is not really sufficient. So we generate this kind of preferred fragments along these regulatory elements. Actually, insufficient fragmentation, actually this is used to identify the regions of regulatory elements in the cells. It's called sono-seq. But sonicate, and then you sequence, and then you get this kind of peaks, for example, along the genome. This correlates with the POL2 binding. But if you use this kind of input chromatin for control, then this will not work. So for most time, then you should really have sufficient fragmentation in order to have an even distribution for input. And then after you get your chromatin input, then next you want to do the chromatin immunoprecipitation. This is the most important step to get high quality data in chip-seq. Then the most important consideration is the antibodies. And this is the key, really. So you really need high-quality antibodies and to get good signals. And then there are no better ways than just testing, validating the antibodies. So just test many antibodies and then choose the best one. And so after you have your antibodies, then what are the controls for the experiments? And so the controls, and usually input chromatin can be used, and also some people use pre-immune IgGs. But the most part, I think the input chromatin is a better control. The reason is that IgGs have a very low affinity for DNA. So for the experiment, you put down very little DNA. And then if you have very little DNA, then you amplify it for library preparation. So the unique reads usually is not sufficient to cover the entire genome. That's why I think the input chromatin is preferred as a kind of a negative control. And also, if possible, try to use different antibodies as control. And also, to control the antibody specificity, whenever possible, using knockout background. So once you get the chromatin DNA, the cheap DNA, the next step is library preparation and sequencing. Now there are standard kits to do this, but only, so this is fairly standard molecular biology techniques. There are not many problems, but one attention should be paid is don't over-amplify your library. Otherwise, you lose the linear range, lose the quantitation. So try to use minimal PCR amplification conditions. And also, once you amplify, then you want to isolate the amplified products, then try to avoid adapter dimers when you're isolating the fragments for sequencing. So once you sequenced, and then the first question is that whether your library, the ChIP-seq works or not. And so usually this is pretty straightforward to test, actually. I think the best way, or not necessarily the best way, but the easiest way is to just display your data in the genome browser, then visually see it. And for example, these are input samples. You see that there are no peaks. And this is H2Z. And then you see these sharp peaks. And you know that this library, this ChIP-seq works. But the two other testing antibodies, for example, this is one antibody, this is another antibody. This is the signal you see. Apparently, this library is not working. So there's no need to do further analysis of this. I reviewed papers that people don't show this kind of data, just show that using the ChIP-seq, then they identified 500 peaks. But I'm pretty sure that if you use any peak calling program to test this, then you would be able to identify hundreds of peaks. And so actually these are not really true peaks. So showing that genome browser display is the first step to see basically whether it works or not, if the data quality is good or not. So once you have the data, then I'm going to only briefly touch on the data analysis because Shirley will extensively examine these aspects. So basically you apply peak calling programs. Then there are numerous peak calling programs nowadays because every computation biologist is smart and want to develop his or her own program to do this analysis. So it's the same thing with the other kind of genomics data, that which one is better, which one is good, which one works for your specific purpose. Need to be really careful about it. And then after peak calling, then it's basically the information you want to see is nowadays usually is the difference observed between the different states or conditions. It's true difference or just kind of experimental artifacts. So just this can be considered, for example, in one of our previous analyses when we compared hematopoietic stem cells with kind of differentiated cells. And if we just plot the average profiling, for example, this is the red is stem cells, and the green is differentiated cells. You see a dramatic difference between these two histone modifications, and also a dramatic difference between another histone modification between the two cells. But So this could be true, but it could be also caused by the experimental, by background errors in the experiments. So after, because these are major histone modifications, we just don't think that there is such big difference between these two cells. So we just assume that maybe the specific rays are just localized to peaks, and therefore we normalize these peaks based on this hypothesis. And now these two conditions basically came together, showing that probably the difference is caused by experimental variations. But to obtain a really meaningful differences between different conditions or cell types, and there are different ways now to do that. Basically, I think these are really, there are several papers reported the different ways of normalization by sparking in input chromatin into the samples. And all this, although the sparking in samples are different, but the idea is the same. Basically, the idea is that once you get, for example, two kind of different conditions, like you get two sets of different peaks, they look dramatically different. But this could be true, could be also experimental variation. So the way to solve the problem is you compare with the controls. For example, if the yellow bar is control, if they are same, and then that's maybe true difference. But maybe if the control are different, then these are experimental variation. And so one set of basically this kind of control, sparking control, is to add reference epigenome or reference chromatin. For example, you have a chromatin. You want to compare the two chromatin status, two cell status, or two conditions. And you have chromatin from the cells with no treatment or the cells with treatment. And then you add a reference epigenome that is identical to both these conditions. After mixing these, and then you do ChIP-seq analysis. And then the hypothesis is that for this kind of a reference epigenome, then you should see identical enrichment on peaks or promoters. And then you should use this to normalize the sequence arrays obtained for these two experimental samples. Now, after normalization, if you still see this kind of difference, then that's most likely true difference. So I will stop here, and then Shelley will talk about the data analysis part.
It's a very interesting workshop today. So I would say actually ChIP-seq analysis is probably among the most complicated because as you see from Keji's talk, the whole procedure is pretty complicated and each step could create some variabilities in the final data. In addition, for ChIP-seq, people could be doing this on transcription factors, chromatin factors, or histomark. And every factor could have a different binding distribution. And so the data is a lot harder to compare. And the third is that in terms of ChIP-seq, you always evaluate it in the context of gene expression. So every complication that people mentioned before on RNA-seq also apply here. So we have double the complication here. So in our case, we feel like in terms of ChIP-seq, just peak color alone, a lot of the current peak colors are probably sufficient. The difficulty is what do you want to do after you do the initial peak calling step? And it's actually pretty complicated. And so we have developed this tool called Systrom. And so this is actually coin-termed by my collaborator, Miles Brown. Systrom stands for all the cis element bound by a transfactor genome-wide. So basically, if you do a ChIP-seq, you get a Systrom data. And you can actually access this from our main Systrom website. So the Systrom analysis pipeline, so kudos to the Galaxy team that Taylor Group have pioneered. So you can actually upload the data. So all the analysis functions are on the left, and you can click on a function to specify the parameters, and all the data, including the initially uploaded data and the processed data, are in the right, and you can specify, I want to use this function, these parameters to analyze another data, and it can be analyzed on the web, and you can look at it, download, and do further analysis. Sorry, it's not showing very well. So we can do a lot of analysis related. I'll just read it from here. You can do chip-seq peak calling. You can check for data reproducibility. You can visualize the signals across different elements. You can look at evolutionary conservation, lift over between genomes, annotate the nearby genes, do motif analysis, draw heat maps, and also integrate with gene expression. And then, I can't read this, sorry, there's some conversion problem. And also, this tool is compatible with genome space. So some of the analysis results, if you load over to other genome space tools, which are connected to gene patterns, to cytoscape, to Galaxy, you can do a lot more analysis. And currently, we have over 6,000 users and 64 terabytes of user data. One thing I mentioned is for chip-seq, it's actually pretty difficult to do integration with transcription gene expression analysis, because if you do chip-seq analysis, very often the binding on the promoters is only 3%. So it's actually very hard to say which one is exactly the real target gene for like transcription factors or some chromatin regulators. And so we have taken this gene-centered approach. So for every gene, we say, okay, it seems like a lot of the other binding sites are all potentially regulating this gene. And the effect is, theoretically, we should get this from Hi-C, although the current resolution of Hi-C is not that great yet. And so we use a more generalized approach. We just say, if we look at this gene's transcription start site and see how many binding sites are nearby, we can weight them by an exponential decay. This decay function is mostly modeled from the existing Hi-C data. It's a general trend, although we know in real life a lot of data could have more specific interactions. But basically, we do this distance-weighted sum of how many binding sites are near a particular gene. And so we believe that the direct target would be those genes. We use this rank product of what we call this regulatory potential, which is this distance-weighted binding and the differential expression to identify the factor's direct target. Because most of the time when people do a chip-seq experiment for, say, a transcription factor, they also have gene expression data when the factor is active and also when the factor is inactive. So you always have differential expression data. And so you can integrate the binding and the differential expression together to get a rank-ordered list of target. Basically, if this gene has many binding sites and the binding sites are close to the gene and this gene is differentially expressed, you're more confident that this is a real target. Of course, when you knock down a factor, there are always differential expressions. So just because some genes are downregulated doesn't mean that this factor is directly downregulating expression. So we ask, compared to all the other down-expressed genes, or all the genes, are the down-expressed genes having more binding sites nearby, or whether the upregulated genes have more binding sites nearby. And so that can actually help us evaluate whether some factor might only do upregulation as its main function, whereas some other factors might be doing both up and down regulation. And interestingly, what we found is that when a factor can have both up and down regulation, it's usually because it's interacting with a different partner. So a motif analysis near those up and down genes on the binding sites can help us figure out what is the factors activating collaborator and the repressive collaborators. And so we have an algorithm called a beta, which is binding and expression to really identify the target. To do all of these analyses, we realize it's actually hard for people to really get all these in place, so we implement that also on the Systrom Galaxy interface. And of course, if you have questions, no tool will be built perfect. So we try to kind of use these Google user groups to continue get feedback and try to fix problems. And also, nowadays there are a lot of public ChIP-seq data available. One of the way for reproducible data analysis is to really collect the data and process them in a uniform way. And so we have also created this resource for the Systrom data browser or the database. So the idea is we download the whole geo, and also we collect the PubMed. We figure out which ones are ChIP-seq data. We download the data, and then we develop a auto parser to parse out the metadata. And then we also use manual curation to double check those sample annotations. And then we process the samples with some standard pipelines, then to try to find relationship between the samples. We hope this will be a useful resource, but this is really very labor intensive, even with all the automation script written. So, when we download all the geo to parse out all the ChIP-seq data, metadata is actually very, very tricky. And so in the manual curation, even just to optimize the process to make the curator's job easier, take some software implementation. We can check all the change histories and try to add more conditions, or make changes to the previous curation. We try to make this process also as automatic, as dynamic as possible. And so with the metadata, we can run some pipelines. We call this pipeline Qilin, which is a pretty comprehensive ChIP-seq quality control and analysis pipeline. Initially, we want to just evaluate how good the ChIP-seq like a read situation is. Is the library complex enough? Do we get good quality reads? Then we do peak calling to figure out, are we getting reproducible peaks? What are the enough fold, good change, good peaks? And then we do annotations to look at evolution, to look at gene expression, and so on. And so this database is available also from the Systrom website. You can search and you can also browse for some human. So these two you can search either by the cell line or the tissues, or you can search by the factors you are interested in. And we have, let's see. So totally auto parse is 20,000. So these are the available ChIP-seq data in the public domain. We have curated 14,000 and processed 10,000. And these are available. So in terms of the initial ChIP-QC measures, we want to make sure the sequencing quality from FastQC is good. The reads are mostly mappable. It has less redundant reads, because if you have a lot of redundant reads, it means the starting material might be too little, or maybe PCR is amplifying too much. We also want to make sure there are enough good peaks with both good false discovery rate and fold change, because in early days when people only sequenced 10 million reads, they may get only 5,000 peaks, and now people are sequencing like 100 million reads, and they're getting like 100,000 peaks. And so when you are sequencing very deep, using p-value or fold change in addition to fold change might be a better cutoff to really decide how many peaks to call. And so for us, if we have too many peaks and the sequencing is too big, we usually use a 5-fold to cut. At that level, we can see that the number of peaks is no longer increasing with sequencing depths. We also evaluate the signal to noise. These are how many reads are in the peaks that you call. And this is, ENCODE call this a FRIP score. And then we use this, the number of peaks that overlap with publicly available DNA's hypersensitive peaks, because if a ChIP-seq is working, especially for transcription factors, it should overlap with DNAs by about 70 to 90%. And also for transcription factors, you should see an enrichment of the motif from the ChIP-seq data. And also the peaks should have a pretty good evolutionary conservation. So all of these are available in this pipeline that you can actually see the QC. For example, if we look at all the estrogen receptor ChIP-seq data here, and we click on a specific data, you can see this is a pretty good data set. All the QC passed the pass. So every tooltip here, if it's red, it didn't pass our QC. If it's red, it's green is good. So the more green, the better. And you can also evaluate the specific QC values. And then we also use this regulatory potential to figure out what are its putative regulatory targets based on how many binding sites are near this gene and their distance. We are currently in the process of integrating this data with GTAX and TCGA gene expression data to really get the better targets. And you can visualize this result from either UCIC genome browser or the WashU Gateway genome browser. And finally, well, we have this analysis. If you are interested in one data, it's like a Netflix. If you're interested in this movie, you might also be interested in other data sets, so you can see what other data are similar. So these are the whole collection of our computational solutions for ChIP-seq. I want to say this is not really a solved problem. There are still many open problems, especially the target analysis and also epigenetic. I think a lot of chromatin regulators, people just don't know how to analyze that data very well. And also ChIP-seq experiments are noisy, and also very few of such ChIP-seq data have replicates. So I really want to, compliment ENCODE. They are generating really high quality data, and all the data have 2 replicates, which I think is very, very important, especially if you're interested in doing differential ChIP-seq calling. I want to mention that if you just do one single replicate between 2 conditions, you might cause some peaks that are differential. But as Keji mentioned, is that real? You might do it again because this experiment is pretty noisy. And the good thing is gene regulation is fairly robust. In the early days when people get 3,000 ChIP-seq peak, their targets versus now the 8,000 peaks actually have been pretty robust in terms of the order of the target. So if you're off by a few thousand binding sites, no big deal, which is actually surprising. But if you're really caring about a specific gene, is this a real differential gene? I would say, yeah, nearby you would probably see multiple binding sites that are differential. And if you have replicates, you'll get more robust result. And as I mentioned, ChIP-seq analysis is quite robust, and it creates challenges to algorithm compatibility because we always have to analyze ChIP-seq with a gene expression, and depends on whether this expression is a growth-seq or an RNA-seq or a microarray. It's hard to make an algorithm that works for all. So in order to have a reproducible data, I would say, for a ChIP-seq paper, the analysis pipeline, the parameters used, and also the overall data quality should be reported in a paper submission. And also, for any big data papers, there are papers that have like 100 ChIP-seq data that's generated. People know that these will be highly cited, and usually journals, like high-profile journals, like these papers. But I think those paper authors need to be more transparent and also open-minded to any challenges about how they actually are doing the analysis, because even though their data quality is good, sometimes not all the analyses are done correctly. So I want to acknowledge members of my lab for making the system, the tools and the databases. We also have a lot of collaborators, including the computational ones and also our experimental collaborators, and many, many collaborators who help us make this happen. And I want to thank the funding agencies. Actually, NHGRI helped us develop the pipeline, and NCI helped us to develop the database, and we're trying to kind of connect them together. We also have some internal fund from the Farber. And yeah, so thanks very much.
Okay, so, and just before I turn it over to questions, I'll say, very briefly two reports. I don't think I have very much to add beyond what the other editors have said. One, two things I would want to emphasize or highlight, what Natalie mentioned, the reporting checklist that the Nature journals have been implementing over the past year or so, I think certainly has helped raise the level of reporting to hopefully make both authors and reviewers aware of some of the things that need to be stated, explicitly, and that you can't assume readers will be able to infer, which includes things like what statistical test you used, how many replicates you did, what the error bars represent. But that's just a tip of the iceberg. Really, it covers a lot of other issues. The other thing, with respect to reproducibility, I would want to mention, some of you might be aware of a fairly recently launched Nature publishing group journal called Scientific Data. And that is essentially a journal that is designed to report large data sets. So the point of those are not, on the analyses that are done on those data sets, but simply a structured way to actually describe your data, describe how it was generated, and also pointing to how the data can be accessed and so forth. And that's a separate publication that is in parallel with your, the publication that might be the analysis of the data. But again, it gives credit to the data producers. It's citable and stable. So that certainly encourage you to kind of have a look at scientific data and the data descriptor format as a way to be able to. Oops, I think I just did that. Again, address this issue of making data accessible and usable for people, in a kind of a stable long-term way. So with that, now I'll just open the floor to questions to our speakers.
What we saw now is that basically the biological data is stochastic, the experiment is stochastic, and more in computational analysis, we're also using stochastic methods. Therefore, in some way, what I would like to ask the researchers, the experts, and also the editors, what's really at the end more important is for us to reproduce exactly the computation pipeline and come back to the same results, or rather describe it in a way so that an expert, be that computational, be that biological, can understand exactly what was done and what could be the pitfalls. So I was a little bit concerned about this, emphasizing on having published the code, which somebody will look, and other than computational experts, nobody can understand, versus having and describe really clearly what was done, and understanding that at the end we have to reproduce the biological results, and not, as the whatever 100 site miss doesn't matter so much. So I was wondering what's your thought about it.
Indeed, I think some of these big data papers, if you were to do the experiment yourself, actually a good ChIP-seq data between replicates, for transcription factors, it's not unusual for the peaks to only overlap by 70%. Actually, that's 80%, 70%. That's very decent data. Although, of course, the more confident peaks usually overlap better. But I think in a way, if other people cannot reproduce your peak call by 100%, that doesn't mean it's bad. But I think the overall conclusions needs the observations. That needs to be correct. So in a way, the data analysis, especially for these bigger studies, the supplementary material really needs to be in much more detail. How exactly is done? We've seen papers where one supplementary material point to, we did the analysis as we did in the last paper, and that paper point to another paper, and that point, and you just never know what is being done. It's like a detective work to figure out what is really happening. And I think in a way, since it's supplementary, just copy and paste, that's still helpful, right? You just need to know how the analysis is done. And maybe the journal or reviewers should be more strict, requiring these more detailed analysis and parameters.
So I agree that clear description of all the methods, either experimental or analysis, is really absolutely required for reproducibility. So I think that nowadays it's good that we can put a lot of words in online, so we are not limited anymore. So there's no excuse basically to not describe clearly.
Yeah, and I can just say that that's true, that our journals, probably many journals now, have relaxed limits on word counts on method sections, and certainly supplementary information has limits to some extent, but more on file size than on word counts. Though I think there is still a balance. I think that there is with consternation. The task that reviewers face when they're asked to review a paper that has 100 pages of supplementary information. I think there is a balance, and I think the burden is on the authors to provide all the information that's necessary, but provide it in a digestible way that will make the task of the reviewers and editors manageable. So, yeah.
Hi. A lot of the writing workshops that I go to for papers emphasize the issue of plagiarism, even copying your own methods into another paper that you're doing. So if you have to have these extensive method sections, how do you deal with the concept of plagiarism within your own work and yet still be entirely detailed with something that you've done similarly to something else.
It's funny, I had the same thought in my head when Shirley talked about cutting and pasting, because there is this issue of self-plagiarism. I think it's one of the smaller, sins of, I think self-plagiarism, for the purposes of providing, information so that the reader doesn't have to trace the paper trail back is, in my mind, not objectionable, but I think, there is this issue that, in scientific integrity, we do take the question of plagiarism as something that we can't ignore. So, I think it's perfectly valid to refer to prior publications, but often there's additional details that should be added, and that can be written in a fresh way, so you're not, copying and pasting exactly, but.
Just as someone who has to deal institutionally with these issues, I think the easiest way to handle this is just to put in the complete description of the technique. If it were published before, indicate as previously published, so that the self-plagiarism is acknowledged, that you're copying yourself, and then add any additional details. And then no one can, accuse you of using work you've already published before, because you're crediting it.
Michael, your point is a good one, because there's that sort of gray zone between citing what you did before and saying what you're doing specifically. As I think James had pointed out, there are circumstances where people have their pipelines and they make minor modifications or tweaks, and unless you know exactly what that is. So, there is this space-saving issue of you can just say, reference 47 is the previous paper, but that's probably not good enough. I mean, you need to be able to say, well, we did it left-handed from the right side of the page, as opposed to, whatever is being modified. I don't want to embarrass myself computationally, so I'll go spatially. But I think that's something that we have to think really hard about, how to encourage people to do that. I mean, editors are always pressing us to decrease the amount of space and say, gee, get it down to 2,000 words. And Kyle, you're my buddies at Nature Genetics, we send long methods, and they say, well, do you really have to say all these things? We've had that. More than one time. More than one time.
So what Shirley mentioned earlier kind of concerned me, what the standard in the field in terms of replication, because you said in general, ChIP-seq data, very few replication. You mentioned that ENCODE data have more. So what is your feeling, both of you, how many replications should be included, biological and technical? And I think this is important in relevance to data reproducibility here.
So I would say, if you only care about roughly where this factor bind, one replicate is probably okay if your antibody is good. I would not recommend doing technical. What is a technical replicate in ChIP-seq? I mean, I think biological replicates, you grow the cell separately, you do a cross-linking sonication, everything, right? That's I think a good replicate for a ChIP-seq. But if you want to compare differential, replicate would be super important. Otherwise, most of the calls will probably be wrong calls.
Okay, well, when you say, technical replication will be using the same sample, but you do the same.
You probably don't need to do that times. For ChIP-seq.
But when you say, okay, that's based on, your data analysis in the past, so what's the basis for
this? Is like, early days when people do ChIP, they get 3,000 peaks. ChIP-seq, they get maybe 5,000. Now with, deeper ChIP-seq, they get 20,000. Most of the target in terms of the ranks, like if you rank it based on this whatever number of binding weighted by distance, they are pretty consistent. So I would say gene regulation is pretty robust. And so a factor, there is no, as Keji mentioned, there is not a very clear yes binding, no binding. It's just more binding, less binding. And so most of it is fairly robust. But if you want to do like a condition-specific or cell-specific binding, then I think 2 replicates would really be needed to call any peak robustly as differential.
So I think two replicates at least always needed. Even the antibodies are good, even just to know where they are, just because human errors can be avoided. Because if you just only do once, then if even the peaks are beautiful, then you don't know whether there are human errors involved. You may use a wrong antibody or use something. So so it's very important to replicate the experiments with the biological replicates, basically.
Time for a couple more questions if anyone has. I will add then, as we wait, see if there are any other questions. I think, the last several, talks and sessions have really focused on some of the technical things that can go wrong in data generation and analysis. And I would want to maybe bring us back again to this issue of study design and statistical framework for evaluating your results. And I think it certainly applies to the first session, for the clinical interpretation of, the sequence variation. But also when it comes to applying, RNA-seq and ChIP-seq, to address questions of biological interest, you still have the conclusions that you can draw from the experiment are going to only be as good as the way you design your study, and that includes what tissues you choose to analyze, and often having a biological question in mind, rather than just doing an experiment hoping that the conclusions and the insights will emerge from the data. Sometimes they will, and I think that's part of the beauty of data-driven science. But there's a balance too, that that there is thoughtful study design applies to the application of genomic sequencing, but also RNA-seq and ChIP-seq. And if there are no other questions, I will turn things over to Stephen Chen.
While Steve is making his way to the podium, first of all, I wanted to thank so many people involved today. All of the speakers did a fantastic job of taking very complex information, explaining why this is a really exciting area of science, and talking about the ways in which we can go wrong if we're not careful. And that's exactly what we wanted, and I'm really very grateful. And thanks to the moderators also for helping to focus the discussion. I also wanted to thank Paul Liu, who has been my colleague and really has organized all of these sessions and done a terrific job, and Jackie Roberts, who made all the arrangements and so on. And we're also grateful to Larry Tabak, who's the NIH Deputy Director, for support, and Tara Schwetz actually is involved in this as well. So on that note, I will turn the podium over to Steve, who is going to explain what we heard today.
All right, well, all right. I feel like, the reduced Shakespeare company where they do Hamlet in 30 seconds, Macbeth in 45 seconds. In sitting here listening to what I thought was a fascinating and a terrific day, tried to pull out some of the themes that we heard. And I used a few of the slides that I started with, and I've updated them and changed them, based on what people had said. But the thing that really struck me was this question that Nate Silver has done very well with poker and with earthquakes and volcanoes in the stock market, and sort of suggests with genomics, but really we haven't got the data in place, and that is this issue of how and in what way we really disambiguate the difference between the signal and the noise. And we clearly saw that as sort of the underlying issue here. What is it that we're trying to get as notable findings that emerge out of these technologies that look at hundreds, thousands, or millions of points or near simultaneously? So the reason I was thinking about this book is I flew back from Japan this last weekend, and I sat next to this gentleman who was reading it, and over 13 hours, we ended up having several conversations as he went through different chapters, per se. But it's a theme that I think we need to think about, because when we really think about today, what we were looking at, whether it's genetics or genomics or transcriptomics or ENCODE, you know, or even clinical genomics, it's how and in what way do we pull these things out. And we are challenged with our analytics. We can generate lots of data, but we don't really still know how to interpret all of that data. We can find wonderful points, and we can be very excited, and I agree with Les that the glass is becoming half full, but we know that that glass has all sorts of things besides water in there, and there may be, you know, wonderful bits of gin and you know, Irish whiskey, or there may be bits of bitter things that none of us want to have. And I again come back to this issue of really the arc. We talked about an awful lot of discovery. We didn't talk as much about the clinical implementation, and it's partly because I think we're still trying to stabilize how we're discovering. And the last discussion on the chip was very, very helpful in sort of seeing how there's been a wonderful sort of scientific crowdsourcing that's taken place, both at the analytic level with that very nice set of tools, as well as how the community is, I think, probably more collegially shared and work through these problems in a shorter period of time. And so I really think that, you know, these two myths are still there. One technology can't do it all, and no single study will get us where we want to go. So really, in thinking about reproducibility, what do we really need? And it was very nice to hear what Hillary had to say, and Natalie and others, that we talked, and Kyle pointed this out, stable study design, careful sample handling, accurate technology, but the key issue here is the appropriate statistical framework, which is still probably the hardest question that someone has. When you generate the data, what is it that you're going to apply to that data, which really maps back to the hypothesis, how you confirm it and replicate it, as we certainly heard with the chip and with the RNA-seq, it's really wide open, and what's that measured interpretation, and what's the ability to really look at the data and computationally have portable solutions that can come in and look at it without having to swamp the clouds by moving large sets of data. Because a lot of what we're doing is talking about pulling that signal out of the noise. And then the question of an enhanced kind of review process. Now, this may be very unpopular with the 4 editors who were here. I mean, but as we've talked about this, there are a couple of things that I'm going to float something that may be particularly unpleasant to them. A pushback to the journals, so to speak, because I think the community is voracious about wanting to generate data and how they publish it, but the sort of heard wafts about how to make or how to think about yet more thorough reviews, whether it's with the computational tools, making them available or portable. You know, and here's the really unpopular possibility of, should there be more internal reviews within the journals? So I give an example from my world of cancer, JNCI, the Journal of the National Cancer Institute, having nothing to do with the NCI. It's an Oxford journal that separated 10, 12 years ago. They have a statistician that looks at every single study and has a separate analysis, and they require looking at the primary data and running some of those analyses to be sure that they indeed can see the kinds of things that are being presented. Now that's, you know, that could be quite onerous, but at least it's something to think about. How and in what way do we do this? Do you do it through the journal? Do you do it through crowdsourcing? Do you do it like as talking with Michael in the break in the physics world, where they have spaces where data go and then people look at it and comment and work on that data before the final publication takes place, as sort of an archiving of the, you know, the what I would call sort of the thorough and the fine assessment of that information. It's something as we generate more and more data, I think we at least have to think about this. I'm not prescriptively saying it goes back to the journals, but I at least want to put that question out there. And similarly, what James kept referring to is something that always worries me a little bit, is what's the gold standard? Genie in the bottle, you know, 1,000 genomes, human genome, these are all great places to start. But those of us who've used the Genome Project to go do cancer genomics find that an awful lot of the things are incorrect. They don't look right because they've been done in lymphoblastoid cell lines and various things, and you find that the P53 normal for some period of time was actually one of the more interesting mutations, and the laboratory has now found out. I mean, so we're correcting ourselves, and that's a very important thing to do. But, you know, gold standards, you know, gold is pure in my mind. We still, you know, if we get 85, 90% with only a little bit of fool's gold, that's very important as to whether thinking that's a priori or a posterior an assessment of gold. And then the most important thing is the full data availability. We have a tension between the genome and the phenome world. The genome, I think, can do this. They got this right. They shared it for the initial assessments and the initial tools, but the phenotyping world has a long, long way to go for that. And there are lots of historical reasons why we're going to have a hard time with what we currently have. And that's why there's sort of this divide with the genomic data sharing policy that we should be able to go forward with more open interpretation. Similarly, I think the correlation does not imply causation. That certainly came through. I think laboratory investigation is really needed. Others do not apply. Okay, I mean, the idea that you can use the genetics all by itself to create the entire story has sort of come through in listening to the ChIP-seq and the RNA, that they're taking, it's more than just running that one run and trying to put it through a computational pipeline. That gives you hypotheses, but how you validate and how you look at that information is very, very important. And so I think laboratory investigation should not be lost in this whole discussion. And particularly as we go into the world of looking at exomes and rarer settings, we do that. So comparing technologies, this is a question that I think that Jim had pointed out, the small and big differences in detection. Type 1 and type 2 errors, it comes back to what's your hypothesis? What can you tolerate vis-a-vis the question that you want to go at? And at the same time, what's the portability of those pipelines? We do have a certain amount of black magic in that things can only be run at the Broad on fire hose on a Tuesday with the right-handed switch, and no one else can see how that outcome is. The Broad does many wonderful things, but again, the ability to be able to get that information to how you run it is absolutely essential, and that's sort of gone through everything that I think we've talked about. And it does raise this question of, we're voracious in wanting to sort of combine all the data, but there is a really good statistical technique out there called meta-analysis, where you don't have to necessarily combine everything, but if you're getting close enough to have likes, you can start to put that together in very useful ways. And I think it's something that we should not forget about. It really wasn't discussed much, but sitting here, I was thinking, there are ways to put together RNA-seq that are done on different machines and be sure, that you're going to account for some of those differences. But there's an awful lot that can overlap and can be very useful. And certainly the chip world, as we heard about, 70% binding or 30% or 50%, really does push this issue of combined versus meta-analysis. The big thing that we, in the last 2 or 3 slides here is transparency. We have to look closely at what people are reporting. And we have to have clear descriptors in the methods. And will the confirmation or the validation swamp the system? What we don't want to do is have perfection be the enemy of the good and not be able to progress. And so there is a sort of a proportion of how much is it going forward full steam and how much do we have to afford some of our time to the validation confirmation. And that's a tension that we haven't really quite figured out. We probably need more confirmation, validation, but not to the point of choking or stopping the system from continuing to use what we had. So that's why I would say I wouldn't want perfection to be the enemy of the very, very good at this point. And that's really where we sort of sit when we start talking about reproducibility and sort of talk ourselves into corners. We know that there are these tensions in the field between old and new platforms and the interoperability, between primary findings and replicates. These are things that we live with a certain amount of dirt there. And then between computational pipelines, what works and why and whose hands and how can these standards be there. But the most important one is the last one there. It's between the genome and phenome worlds. We really are worlds apart in terms of the data sharing, and that's going to always be a challenge for the reproducibility. So the question that I tried to ask before in one of the earlier sessions is, should there be intermediate stages where you at least can show some portion of the data that would be able to be looked at by others and be sure that the integrity and the results are stable? That would be an assurance to, indeed, those who are reading the paper or looking at the data, that they could really go forward with that. So with that, I think those are the comments that I'd like to make in sort of hearing whatever, and listening to what I thought was a really spectacular day. And I thank Michael for really starting this series, and Paul and all the others for really make this happen. So Michael, do you want a final word or say thank you and go off and hope the journalists the journal editors don't shoot me for some of the things I said. I have papers coming to each of you at some point in the near future. So be friendly and forget about what I said. All right. Again, thank you one and all. Okay.